chromosome 2 fusion is probably one of my favorite lines of evidence for evolution. It's one of the coolest instances of a logical prediction that was later validated by science. So, to start out with, most people know what chromosomes are. When your genome replicates, it splits off into like tight little entities called chromosomes. And the number of chromosomes varies from species to species. Now, all of the other great apes have 48 chromosomes, but humans have 46. So that's 23 pairs versus 24 pairs in all other great apes. So if evolution was true and humans really did evolve from some kind of ape, then we would expect that at some point in that evolution that humans would have had to have lost a chromosome pair, i.e., a fusion would have had to occur between two ape chromosomes to get to one human chromosome so that overall the numbers work out and you go from 48 to 46 chromosomes, or 24 to 23 pairs of chromosomes that all humans have today. So that's the prediction, that we would expect to see some kind of evidence that a fusion occurred. That is the logic behind why we would make that prediction with an evolutionary model. Now, when that prediction was obviously made a while before we had the technology to actually investigate this. Now, when the actual chromosomes were investigated, something very interesting was found. There was something called a fish analysis, which sounds a little bit goofy, but it's like a fluorescence in situ hybridization. So all that basically means is that they ordered little like proteins that would come along and bind with the chromosome in an order, right? So you'd get A, B, C, D, E. Like there's a distinct order to the chromosome. So it allows us to map, not just genes, but just in general, the genetic sequence follows in that order. And when we compare this to chimpanzee chromosomes, we find that there are two chromosomes in the chimpanzee genome which exactly match. When you line them up end to end, they exactly match the same sequence as human chromosome 2. So the A, B, C, D, E, F, G, all the way, the sequence is the same. When you line up those two chimpanzee chromosomes, the sequence is the same as human chromosome number 2. So this was essentially, when this came out in the, I think, early 2000s, this is enough that basically convinced the science, the geneticist, the biologist. This is basically what got the consensus that yes, chromosome 2 fusion did happen. The analog for this would basically be if somebody took two eggshells, right? You have an egg that's cracked in two, and you match the two halves together, and you find that they perfectly align all of the jagged edges and the grooves. It's a very reasonable thing to conclude, a very logical, rational thing to conclude, that that was the same egg that cracked in two. And it's the same kind of reasoning that we're using, like kind of building a case like forensics style, like a detective case, gathering the evidence. And it's very, very strong evidence when the two chimpanzee chromosomes, when you line them up end to end, they perfectly match the sequence of human chromosome 2 in a fish analysis. That's the nail in the coffin that convinced all the scientists. But since then, we've had even further discoveries, like the sequencing of all of the human genome. And with that, we noticed that there are actually what are called telomeric sequences in the middle of chromosome 2. Now, telomeres are the end of chromosomes. So you only find telomeric sequences near the ends of chromosomes. But in chromosome 2, we find, not only do we find telomeric sequences in the middle of chromosome 2, but we actually find them end to end, kind of like head to head, which is the only place in the genome where we find such an end-to-end sequence of telomeric repeats, which is exactly what we would expect to find as sort of the remnants of an end-to-end chromosome fusion. So it checks out on basically every level. It is, at this point, essentially a foregone conclusion. I mean, all of the scientific consensus, as soon as they did the fish analysis, that's what convinced the scientists. So this is just the icing on the cake. And then the last thing that I will bring up, which is really the nail in the coffin, or the final nail in the coffin in my book, because oftentimes creationists like SFT or like Tompkins will bring up the fact that, oh, there's a gene that's right next to this supposed fusion site. And how could that be possible? This DDX11L2 gene, which I imagine Donnie will probably bring up, or it will come up at some point tonight. So I just wanted to address that really quick. If the human chromosome 2 is a result of fusion from two ape chromosomes, then we would expect that DDX11L2, if it's located so close to the fusion site in the middle of human chromosome 2 now, we would expect that that gene, or a very similar gene, would be found at the end next to the telomeres at one of the two chimpanzee chromosomes that are the analogs for the chromosomes that fused to make chromosome 2. And that is exactly what we find. Right at the end of chimp chromosome 2A, we find DDX11L2, right where we would predict that it should be, right next to the telomere that would fuse in human chromosome 2, which is why we find DDX11L2 right next to that site in our genomes today. So that's a lot. It's a very technical topic, but it is one of my favorite topics. So I'm looking forward to seeing how Donnie can possibly spin this, because these seem like very foolproof lines of evidence that there's, I mean, it seems so obvious that anyone looking at this would have to conclude that a chromosome fusion did occur in the past. So I guess I'll leave it at that. We'll see what Donnie's responses are, but I really can't see much of a way around this. This is like, the fish analysis matches. That's the main strongest line of evidence and reasoning that I don't think that you can get around. So we'll see how Donnie spins it tonight. Thanks.
Thank you so much there, Grayson. You had about a few minutes to spare. Well, let's add that into the open discussion. And SFT, will hand it off to you when you're ready, and I'll start your timer.
Okay, thank you, Praise. Just 1 second. How's my audio coming in?
Sounds good.
All right. I will start my timer in 3, 2, 1. All right, let's get right into this. I'm looking forward to this debate, and I do appreciate Grayson's thorough and well-prepared opening statement. I've organized my opening statement in a way that first covers the details of this topic, and also why this topic has incredible implications for the ancestry debate. So let's start with the basics here. Apes have 48 chromosomes, and humans, whom evolutionary scientists classify in a subgroup of primates known as the great apes, have 46 chromosomes. Humans stand out as the only species within the great apes as having 46 chromosomes, as Grayson pointed out in his opening statement. Since protagonists of evolution like Grayson here believe the great apes, again, this includes humans according to their model of ancestry, share a common ancestor, they have appealed to an imaginary fusion event in the human line. This event occurred at a point in time after the hypothetical split between the human and chimpanzee common ancestor, roughly 6 to 10 million years ago, according to many evolutionary scientists. Basically, the common ancestor for humans and chimpanzees would have had 48 chromosomes. Then, sometime after the split from this common ancestor, the human line underwent a fusion event. And this genetic mistake, or unilateral mistake, resulted in humans today having 46 chromosomes rather than 48. This fusion would have had to spread and become fixed in an early human-like population to explain why all humans today have 46 chromosomes and not 48. Can Grayson give me a plausible explanation on how this was possible? without invoking fanciful storytelling. Defenders of common descent assume humans and chimpanzees are related to a common ancestor and therefore expect to find evidence for a fusion somewhere in the human genome. Biblical creationists like myself don't believe humans are related to the great apes, like the chimpanzees, gorillas, bonobos, orangutans, so on and so forth. I argue that humans were created separate from every other form of life. I also defend the designed diversity model that explains the majority of DNA differences and DNA units as being the result of initial design or creation. God would have front-loaded Adam and Eve in the original created kinds with pre-existing diversity or heterozygosity, and also with many types of functional DNA elements. This model makes very specific, and I would argue obvious, predictions. One major prediction involves DNA function. I argue that most of the genome, in this specific case, the genome of humans, is functional. This includes the many types of DNA units or elements that I debate endlessly with evolutionists over, such as retroelements, pseudogenes, and allele sequences. Why is this important, though, for our debate on the human chromosome 2? Since the debate centers around the ancestry topic, universal or separate ancestry, the fact that humans have 46 chromosomes and our so-called closest cousin on the evolutionary tree of life, the chimpanzee, has 48, gives us one of many lines of evidence that sets humans apart from the rest of the biological world. For the so-called fusion site that we will be discussing comprehensively in this debate, I would therefore predict function for the sequences and genes that my opponent Grayson here tonight will claim, as he has done so in his opening statement, are evidence for an end-to-end fusion of two ape-like chromosomes millions of years ago. Can I demonstrate that the purported fusion site, including the alleged cryptic centromere site, are functional features inside active and essential genes, and not the ancient remnants of a chromosome fusion catastrophe in the past? The answer is yes. These features Grayson points to, as he has done so in his opening statement, as evidence for a fusion event, are functional elements overlapped by highly efficient genes. The earliest hint of a chromosome fusion in the human line, as Grayson touched on in his opening statement, I do appreciate it, Grayson, after, of course, the hypothetical chimp-human split took place, was prior to genetic sequencing. Scientists were looking at images of human and chimpanzee chromosomes through an impressive technique or process called karyotyping. In this method, you can isolate cells as they are dividing during mitosis. The staining that takes place during this procedure gives us a remarkable visualizing of the chromosomes. It generates these bands in chromosomes. Now, based on this practice of visualizing chromosomes and their banding patterns, scientists allege that two chromosomes, now called 2A and 2B in chimpanzees, fuse to form human chromosome 2. For the evolutionist, this accounts for the disparities in chromosome numbers between humans and the great apes. These initial images are what first gave the evolutionary community the notion that there may have been a fusion in the human line. Now, when genetic sequencing technologies began to expand, scientists discovered what they believed was the setting of a putative fusion in the human line. There is a specific DNA sequence that exists at the end of chromosomes called telomeric DNA. In humans, these sequences are approximately 5,000 to 15,000 bases long. Now, if two telomeres fuse together, we would anticipate a sizable DNA signature or mark, at least 10,000 to 30,000 bases in length. What was uncovered in the human chromosome 2 was only 798 base pairs, an interesting observation and reality that Grayson failed to point out in his opening statement. The telomeric sequences found at this site were in both forwards and reverse, as Grayson pointed out. Now, DNA is double-stranded. Therefore, the forward sequence is TTAGGG, and the complement to it is CCCTAA. This so-called fusion signature is exceptionally degenerate. The site is only 70% similar to what a pure fusion site would look like if it were 798 base pairs. The site is incredibly small. It's corrupt. The authors of a famous fusion paper pointed out just how degenerate the site is. And these aren't young Earth creationists. I quote, "If the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate?" That's a question I want Grayson to answer. It is crucial to note that this 798 base pair region, though, is right in the middle of an important gene, the DDX11L2 gene. The evolutionary community has failed to answer the question, how do you get highly functional genes by slamming together two chromosomes? Evolutionists like Grayson want us to believe so badly that we can gain or form highly critical and essential genes by slamming together two chromosomes. This doesn't happen. Some badly informed evolutionists have argued that the gene that overlaps the so-called fusion site, the DDX11L2 gene, is non-functional because they declare it's a pseudogene or a broken gene that doesn't code for protein. We now understand that there are many more long non-coding RNA genes than there are protein coding genes. And these long non-coding RNA genes are essential for healthy cell processes. The protein coding genes are the building blocks, which is one of the reasons why they tend to be more similar among creatures. But the non-coding RNA genes are more taxonomically restricted or creature specific. These genes provide the metadata for the building blocks that are the protein coding genes to properly function and carry out their jobs. Long non-coding RNAs function in a variety of ways. Many of them function in the nucleus. They modify DNA. In a paper titled "Unique Features of Long Non-Coding RNA Biogenesis and Functions," the authors state, and I quote, "Long non-coding RNAs are a diverse class of RNAs that engage in numerous biological processes across every branch of life." Another technical paper titled "Many Human Large Intergenic Non-Coding RNAs Associate with Chromatin Modifying Complexes and Affect Gene Expression" says, and I quote again, "There is a growing body of literature from yeast to mammals suggesting the non-coding RNAs have an important role in chromatin state formation." Now, the fusion site, it is actually read in the reverse complement, and it is a transcription factor binding site in the middle of what we just discussed, a long non-coding RNA helicase gene. It codes for a functional RNA molecule that is functioning in the genome. The site itself is an essential genetic switch called a promoter that controls and regulates the DDX11L2 gene. The site binds 12 different types of transcription factors, which, for the audience's sake, are proteins that latch onto a gene and turn it off and on, or tell it how fast to run and how much product to make. RNA polymerase II binds inside the fusion site and starts transcription. The proposed fusion site turns out to not be a fusion site. It is a key genomic switch within a highly efficient gene. The gene that crosses the alleged fusion location is a long non-coding RNA gene. I've got exactly 2 minutes here. Long non-coding RNA genes have been knocked out in mice, and the consequence has been lethal. This indicates, this tells us they are necessary for the cell. If they weren't needed, they wouldn't have these detrimental effects. They have also been linked with different types of diseases and cancers. Mutating a healthy long non-coding RNA gene produces disease because the gene is valuable for the body. It's essential. This means these functional DNA units are crucial to health and need to be preserved by the cell. The DDX11L2 gene is situated in a region of many other important genes. It exists in a gene-rich area of the genome. Something Grayson didn't cover, and I hope he does when it comes to the synteny and the homology. Well, many, if not all, of the thousands of genes surrounding the DDX11L2 gene are not found in the chimpanzee. Evolutionists have argued that all of these genes, and I wonder if Grayson would argue the same, got to where they are by moving or migrating from other areas of the human genome. This is storytelling. It's wishful thinking, guys. There is no good homology between the respective locations within human and chimpanzee genomes, and that was the majority of Grayson's argument. You can easily check the genetic database for yourself for verification of all this. You don't have to take my word for it. Simply search the DDX11L2 gene. You'll find, and I quote, DDX11L2 has 1,996 functional associations with biological entities spanning 7 categories. I've got 20 seconds here. Here's the last part. The evidence is clear. The so-called fusion site, now understood to be an active promoter or genetic switch, comprises numerous start points for transcription where RNA polymerase II binds. There exist multiple transcription start points within this region and site that the evolutionary community claims are the ancient remnants of a chromosomal fusion. Guys, this simply is not possible. Thank you very much. Praise when you speak, just please be cognizant of your mic. It's on mute.
Oh, wow. It's my fault there. Yeah, I appreciate both those openers were fantastic. So we'll head in now to the rebuttal session, the 8-minute rebuttal session. And since Donnie just went, we'll hand it off to Grayson now. Whenever you're ready for your rebuttal, I'll take your time.
Okay, well, that was pretty amusing, so I'll just kind of go through in order of what Donnie brought up. First, he wanted to know why the telomeric sequences in the fusion site in chromosome 2 are so short and why they're so degenerated. Well, obviously, a end-to-end chromosome fusion is not going to occur if the telomeres are not degraded and shortened. That is pretty rational to think. If the telomeres are little end caps that protect the ends of chromosomes from fusing. So you are not going to achieve a fusion. You're not going to get a fusion unless those telomeres are highly degraded. So it's right in line with what we would expect of a fusion. Now, Donnie mentions, I tried to write down the quotes. He said DDX11L2 was an important gene. It was highly functional. It's highly critical and functional. He continued to make this claim that DDX11L2 was a highly functional gene. Now, this, for the audience listening, is a complete misleading statement. This is bordering on a complete lie or misinformation. There is no known function for DDX11L2. He wants to claim that this is a long non-coding RNA that can affect gene expression and chromatin state, but he has no evidence of this. In fact, no one does. The DDX11L2 gene transcript, right, once it's transcribed by RNA polymerase, has no known function. Nobody knows what it does. Any gene transcript will have the interactions that Donnie read out. He said, oh, it interacts with all of these different sites. Literally any RNA transcript will have an amount of interactions that border on that. That is the nature of biochemical activity, right? These things are floating around at random and hitting whatever they hit and interacting with whatever they interact with the body. Like, this is all how spurious activity works. You can literally look up any RNA gene transcript and you're going to find a very high number of interactions in these in vitro studies. So then, he hasn't demonstrated that this is non-coding RNA. He has not demonstrated that this is a highly functional gene as he claims. Again, this is a pseudogene. That is how it's characterized. Notice how Donnie never mentioned that once. He completely avoided it. In fact, he mislabeled it as a gene, which, like, all of our knowledge of genetics, all of our knowledge of biology, everywhere you look at DDX11L2, everyone will say it's a pseudogene with no known function. If Donnie can propose a function, then he would be able to publish that paper because it would be new knowledge in science. Literally, nobody knows what the function is. It is not a gene. It is a pseudogene. That is how it is classified. So I would appreciate it if he could at least not mislabel it. And then he wanted to say that there were a bunch of genes next to the DDX11L2 gene that are not found in chimpanzees. I'm certainly not familiar with any of these, but if I'm happy to read up on them. But he said, oh, does Grayson really believe that genes can change location in the chromosomes? Like, he's asking this so incredulously, but this is a very well known and documented phenomenon of chromosomal translocation. Like, this is something that you read about in textbooks when you're learning this information. Like, this is standard knowledge. We know that genes can change location in chromosomes. We know the mechanisms by how they do that. And I know that Donnie knows about this because if you ever listen to his discussions on ERVs, he loves to bring up jumping genes. Like, he uses jumping genes for his positions on ERV. So he knows that genes can change locations. That's what a transposon is. But there's other kinds that, like I said, just chromosomal translocations. So yeah, there is nothing that Donnie brought up that is inconsistent with the evolutionary model. Donnie likes to bring up all these things about, oh, it's the fusion site is in the middle of an important gene. Like, there's nothing about that is inconsistent with the model of chromosome 2 fusion. There's nothing about that is a delineating line of evidence that disproves one or is even stronger on one side or the other. Like, we know that genes can change locations, and we know that promoters are very robust, right? Like, the way that he wanted to say that the actual fusion site itself is a promoter, which is like not the case to my knowledge. The promoter is on one side of the fusion, and then the DDX11L2 gene is on the other side. So promoters can also change places in the genome, and promoters can be like created from mutations, like neutral mutations. Like, promoters are very robust. You don't need a specific sequence. Like, RNA polymerase will bind to lots of different sites. Like, promoter is not some big thing that we can't account for or is inconsistent with a chromosome 2 fusion model. So none of what Donnie brought up in any way invalidates or is inconsistent with the chromosome 2 fusion model. And I would like it if Donnie could somehow explain the fish analysis, which shows it's like the same gene sequence. Whenever you line up the two chimp chromosomes, it's the same sequence in chromosome 2. Like, is that just a massive coincidence? Why would God do that? I mean, God is making it look like humans were not specially created and evolved from chimps. Like, when we look at the genomes, that is what any rational person sees. That's what all the geneticists see, except for Tompkins and the young earth creationists that have, very important presuppositions that are near and dear to their belief systems. But like, I don't see how he has a leg to stand on, and I don't see how anything that he has brought up invalidates or is inconsistent with the chromosome 2 fusion position. And he has not been able to respond to any of the evidences that I brought up, like the fact that the DDX11L2 gene is in chimp chromosomes, right where a fusion model would expect it to be, right next to the chromosomes on one of the two chimp chromosomes that fused. So there you go. I mean, I would appreciate it if I could hear some responses to the lines of evidence.
Much appreciated there, Grayson. Well, I think we're going to hand it off now to SFT for his 8-minute rebuttal. Then we'll head into the open discussion.
Okay, thank you, Praise. Let me get my timer going here. I've got a lot of notes. And I appreciate, I'll start my timer now. Got 8 minutes. I appreciate Grayson's passion and energy. This is going to be fun. So Grayson argued that the DDX11L2 is not functional and is basically just spurious or noise. Guys, this site is a transcription factor binding site in the middle of a gene called the DDX11L2 gene, which is an RNA helicase gene. It doesn't code for a protein, yes, but it does code for a functional RNA molecule. This is evolutionary philosophy to look to these specific genes and say, well, this is a pseudogene. This is a genetic mistake. No, again, that's evolutionary philosophy that is assuming that pseudogenes actually represent a gene that used to be a protein coding gene and is now broken. No, we understand pseudogenes are necessary to sustain healthy life processes in the cell. It is highly co-expressed with other critical genes. And these genes are critical to the cell cycle and signaling in the extracellular matrix, which is the area outside your cells where a lot of the cell communication goes on. Okay, guys, there's paper after paper talking about how the genome is akin to an RNA machine. And the DDX11L2, Gracie keeps saying, we don't know what it does. Nobody has evidence for what it does. This gene produces long non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. And the second promoter or genetic switch of this gene is the fusion site itself. Now, it sounds like Grayson is using a similar or a modified argument to what Ken Miller has said. And he's argued that the purported fusion site is 1,300 bases away from the DDX11L2 gene. He has been proven wrong on this. It's on record. According to Dr. Jeffrey Tompkins, who has had an extensive email exchange with him, Ken Miller has admitted to being in error. The site is in the middle of the first intron. The evidence is clear, and evolutionists deny the evidence. This area in the genome, it's not a fusion site. It is a functional DNA element that is highly expressed and necessary to sustain processes in the cell. So critics who continue to repeat this pseudogene argument are simply not up to date on the technical literature. Now, Grayson, in his opening statement, he covered details on what chromosomes are. I appreciate that. He covered what I covered in my opening, the number of chromosomes in humans versus the great apes, or non-human great apes, as evolutionists would say. He discussed the prediction of a fusion and argued that the prediction was fulfilled. Is this really the case? Well, for sake of argument in this debate, since I am specifically arguing there was no fusion, let's assume that that is true. This was a strong prediction. Well, if there was no fusion, which the evidence supports, then this was a failed prediction. Grayson talked a lot about the homology and the synteny between human chromosome 2 and the chimpanzee 2 chromosomes, 2A and 2B. Is what he is saying is true? The answer is no. And I'll explain why, okay? So firstly, yes, 600,000 bases of DNA that surround the so-called fusion site have been sequenced, and researchers discovered that the gene, this DDX11L2 gene, was nested with many other important genes. The problem for the evolutionists is that none of these genes within the 600,000 base region are found in the chimpanzee. Where did they go? It has been hypothesized, and it seems like that is what Grayson's going to hold to here, that all the genes managed to get to where they are by migrating from other places within the genome. Again, this is wishful thinking. It's storytelling. Evolutionists are so good at it. The synteny argument. Well, firstly, synteny and the 3D organization of the genomes of biological organisms is best explained by the common design model. Since the way genomes are organized and the ordering of genes within chromosomes, there's paper after paper on this, are significant for functional reasons. Chromosomal order greatly influences the way genes perform. The elegant and sophisticated nature of the genome is powerful evidence for homology and synteny by design, not by what Grayson is pointing out here, okay? Homology and synteny exist for functional reasons, but when specifically Dr. Jeffrey Tompkins, a PhD geneticist, he looked into this claim of synteny and he concluded no evidence of synteny with chimp for the purported fusion site was found. The 798 base core fusion site sequence does not align to its predicted orthologous telomeric regions in the chimp genome on chromosomes 2A and 2B. What about the high density that he talks about, these internally located telomeric repeats? Well, firstly, the human chromosome 2 is actually the second biggest chromosome in the genome next to the chromosome 1. So these evolutionists had to pick a chromosome to say was fused at some point to save the day. So they chose the chromosome 2. Again, it's a large chromosome, but these interstitial telomeric repeats, they are located on all chromosomes. Some chromosomes, they actually contain almost as much that we find on the chromosome 2, and they have nothing to do with a fusion. They're there for gene expression, functional reasons, okay? The fusion site region, again, 798 base pairs, comprises multiple start points for transcription where RNA polymerase II binds. It's an amazing source of genome activity. So yes, we are going to expect some uniqueness associated with the chromosome 2, but the uniqueness is not because of an ancient fusion event. It's because of what it's doing, its activity, its size. The evidence firmly indicates that this location, again, it's not a fusion, it's a failed prediction. Okay. And when it comes to the internally located telomeric repeats in general, it turns out, and I've examined all the chromosomes, I've looked at where these telomeric repeats are located, and in the chromosome 2 fusion site area, very few of them, of these repeats, actually appear in tandem. The sequence of the 798 base pair region contains only a few occurrences where 2 repeats are actually in tandem. We should find hundreds to thousands of these repeats in tandem. And there are none that have 3 repeats or more. And there's many of these interstitial telomere sites all over the human genome where the repeats, guess what, occur in perfect tandem 3 to 10 times or even more. And so there's other chromosomes that have uniqueness to them when compared to the chromosome 2 fusion. So these guys, they want to cherry pick the data and say, well, what we're looking at here in the chromosome 2 is indicative of a fusion, but over here in these other chromosomes where we find the repeats occurring in perfect tandem 3 to 10 times or even more, when at the fusion site you only find it a few times, you know, they want to ignore that uniqueness. Well, again, internally located clusters of telomere repeats, as Grayson points to here, are much more frequent than is recognized and not automatically indicative of a fusion. Literally thousands of telomeric repeats across the genome have been directly associated with hallmarks of gene expression. What was my prediction in my opening statement in my last 30 seconds here? It was function. If we can demonstrate that these features within the chromosome 2 are functional elements within functional human genes, then that overturns the chromosome 2 fusion argument. The most parsimonious explanation is this is there by design. And the same type of transcription factor binding and gene activity happening at the alleged fusion site were occurring genome-wide at numerous other interstitial telomeric repeats. Guys, we're looking at designed functional code within human genes, and that's 8 minutes. Thank you.
Much obliged, SFT, and thank you guys for your rebuttals there. We want to make this open discussion smooth as possible so we can just jump right into it. So, and maybe if Grayson just wants to respond to that last point, you guys can continue that conversation.
Yeah, definitely. I just want to ask Donnie, can you acknowledge that chromosomal translocation exists and that genes can change locations in the chromosome, and it actually happens pretty frequently?
Good question. And I would argue that since jumping genes do exist, I've done a lot of study on them, that they can move from one chromosomal location to another. It does happen, but again, these are genes within 600,000 base pairs that surround the so-called fusion site, the 798 base pair site.
Yeah.
Those genes are important. They're critical. And none of them exist within the chimpanzee. So how did they get there in the human area within the chromosome 2 fusion? You're saying that they all migrate? I get migration happens. There are jumping genes, but you're going to have to invoke a lot of movement and translocation.
No, it can just be one migration event, like a 600,000 base pair segment of the genome can translocate, like during recombination or various like chromosome like formations. Like the mechanisms behind chromosomal translocation are known, and entire chunks of the chromosome can move.
Can you point to me another area in the genome on any chromosome where we have something similar, where we literally have 600,000 bases of DNA? And these researchers, I can send you the paper after, they sequenced all of those bases that surround the DDX11L2 and demonstrated that the DDX11L2 gene, it's nested with many other critical essential genes. And none of those genes, none of them within 600,000 bases are found in the chimpanzee. Can you show me something similar where this type of migration and translocation has occurred in other genes and other chromosomes?
Yeah, I'm not familiar with this study. Was this done in a secular journal, or is this creationist literature like with Tompkins? Because to my knowledge, like there really is not a lot of secular literature on the DDX11L2 gene.
Good question. It's in the secular literature.
Yeah, okay. So yeah, I can't provide examples of chromosomal translocation, like segments. I don't have that information offhand. But again, you can look it up in any genetics textbook. Like chromosomal translocation is not incredibly rare. I mean, it's common and understood procedure that like we can
Well,
document.
just because somebody can bench 200 pounds doesn't mean that they're going to be able to bench 1,200 pounds, or I can jump and one day I'm going to jump over the moon. So yeah, translocation happens. Jumping genes exist within the genome where they can move from one chromosomal location to another. But that doesn't mean that you're going to be able to use those mechanisms now. And to me, that's an extrapolation fallacy. And now say, well, all of these important genes within the 600,000 bases surrounding the DDX11L2 gene, they just all migrated there and managed to become essential. And we got this beautiful nesting within the DDX11L2 gene. I mean, sure. But to me, that's a lot of evolutionary philosophy, storytelling. It comes down to parsimony. The most
Well,
parsimony.
I didn't understand what you just were talking about here with like the nesting within the chromosome 2 genes or how they became functional. I think that's a little bit of a straw man. I mean, I'm not proposing that any of these things became functional after a
Okay, so you're
translocation.
saying functional genes, pre-existing functional genes in the human genome migrated to that area, and so they were already functional, you're saying?
Yeah, and we see this happen. That's another thing. You claimed that like the order and location of these genes is super critical and important, but we've seen translocations happen that don't impact pretty much anything. Like you can change locations of genes. Like some genes are going to be more location dependent than others, but this is not a blanket statement by any means. Some genes are not location dependent. They can be on anywhere in the chromosome. And again, when you were saying that somehow like the location is necessary to the design and that's why the locations and the order are the same in chromosome 2 to the two ape chromosomes, but like that falls apart on the face of it because some of the genes that need to be in a certain location in the chimp chromosome under your view are now all of a sudden, instead of being near the telomere, now they're in the middle of chromosome 2, and now you've totally changed their location from near the end of a chromosome to in the middle of a chromosome. So like in your model, like I mean the location is changing. So
Well, let's do one point at a time. I do appreciate that. My response was to your homology argument, and synteny argument, right? Shared gene order along chromosomes. Firstly, I pointed out that synteny, I've got at least 6 papers here that show that the existence of synteny in chromosomes across different species is there for many functional purposes. But the homology itself, this entire region of the human chromosome 2, based on many different people's analyses, they've pointed out don't appear to have any homology with any part of the 2 chimp chromosomes that are thought to have fused. So I would agree with you that there's some similarity there. There's some homology, there's some synteny, but I feel like you are mainly focusing on the sum, but ignoring a lot of the dissimilarity. One
but what
being what
about
we
the
just
fish analysis? In the fish analysis, which was the main paper that demonstrated chromosome 2 fusion, there was not even any disagreement between the order. Like it was 100% the same. There was no difference in the ordering of like the fluorescent proteins in the fish analysis. There was not any difference
now
between the 2 chromosomes of the chimps and the one human chromosome 2.
Now, correct me if I'm wrong, this fish analysis, is this actual base by base, looking at the thousands of base pairs, comparing them, or is this the karyotyping, basically, where you take
It's neither. So the fish analysis is not, it's like sort of indirectly looking at the sequences, but this is before like genetic sequencing was like cheap and widely available. This is like in the early
Right.
2000s before the human genome was sequenced. So
Right.
fish analysis is where they take fluorescent proteins which will like preferentially bind to certain areas. Like that they will look for certain sequences and they're going to reliably and consistently bind with some sequences more than others. So when you release a whole bunch of different ones, they will bind in a certain order all along human chromosome 2, right? Just based on the sequences. So then you can just read, because these things are glowing. These are fluorescent proteins. So they're glowing in different colors. Like we can read the order. Like you're reading, like in my example, I did A, B, C, D, E. Like that's the order in human chromosome 2. And then they introduce the exact same
It's shared gene order, right? You're saying
it's not just gene order. I was specific about that. It's not just binding to genes. It's just binding to areas of the chromosome. So it can be coding, non-coding, look, whatever. They're just binding to where they're going to bind with in a certain order. And it's the exact same order, not even one single difference in the order from human chromosome 2 to the 2 end-to-end ape chromosomes lined up together.
Okay, so firstly, there's a couple issues. And whether it's homology, whether it's synteny, I am pointing out that they are there for functional reasons. So it's not going to do much in terms of differentiating our positions. I would say that the 600,000 bases where you've got all these genes and then the DDX11L2 nested in, I would argue that that's pretty close to a differentiating line of evidence, but you're going to say, and we'll move on from that just to close that topic, that these genes migrated, translocated, they got there from other places within the human genome. Okay, I would argue the most parsimonious explanation is the reason why none of these genes within this 600,000 bases of DNA is in the chimpanzees, because they never were there. But with what you're saying is, when it comes to looking at these comparisons, there's over 48% of chimpanzee chromosome 2B for, you got 2A, 2B, which allegedly contributed to what we're looking here at the fusion site and the cryptic centromere. They're actually nothing but meaningless ends with just thousands of gaps. And you can actually find one really large notable gap covering nearly half the chromosome. And the reason why is the assembly of a lot of your chimpanzee chromosomes, it's not been highly sequenced. So there's a lot of meaningless data. And so where you're looking at synteny and shared gene order and non-coding regions and so on and so forth, there is a lot of guesswork, because the assembly itself, and I'm not sure if you're aware of this, if you can speak to this a little bit, unfortunately, that there is a lot of data there that we just don't know. There's a lot of inferring based on the human genome being used as a scaffold. Speak to that a little bit.
Right. In the fish analysis in particular, there's absolutely no assumptions about that, right? Like you are literally looking at like the staining. You are seeing the order. So there's no assumptions. Like it's not like you don't even need to know the base pair sequences in order to do the fish analysis. Like you can just read it with your eyeballs, like what order these proteins are binding and staining the chromosome.
Right. But again, if you're looking at specific genes and proteins and you're comparing chimpanzee chromosomes 2A and 2B to the human chromosome 2, there is still going to be a lot of meaningless data, at least in the chimpanzee chromosomes, because not all of that has been sequenced fully, or there hasn't been enough.
It doesn't need to be sequenced at all in order to do the fish analysis.
Well, then you're not really going to get a incredibly sophisticated analysis then, if it seems like you're leaving a lot out
there. I don't understand why that follows. I mean, like the proteins like that are staining the chromosome in the order are binding specific sequences. Like it doesn't matter if we know them or not, they're going to bind the same sequence in like the human chromosome and the chimp chromosome.
Okay, well, that's fine. If we do see some similarity there, and it's a bit of synteny, it's a little bit of homology, we're looking at different proteins, the staining process, I would argue again that all of these similarities, this is not going to differentiate the evidence within this debate, because that could certainly be there for, again, functional reasons. Like we know the synteny exists. I'm specifically interested, and I'm currently looking at it, I think you've looked at it, all your chromosomes within humans, and basically these internally existing telomeric repeats, and where they're specifically located on the chromosomes. And so if you're familiar with it, if you look to human chromosome 2, there is, I'm not sure if you've seen these specific images, but there is the highest density of these internally clustered telomeric repeats at the chromosome 2 fusion site. Okay. Speak to that a little bit. How does that support your position? And maybe we can go back, because I think this is going to be one way to differentiate the
So
point
yeah,
for sure.
the
Go ahead.
key with like the interstitial telomeric repeats, right, is that like sure you can find instances of those throughout the genome, although yes, there's a high amount of them in more some places more than others, such as in the middle of chromosome 2 where the fusion site is. But there's other places you can find interstitial telomeric repeats, like they're all over chromosome 9, for instance.
Right.
But the main key is that these telomeric repeats are in an end-to-end, a head-to-head formation in the middle of chromosome 2. And there's no other place in the genome that exhibits that sequence. That's unique to the middle of chromosome 2.
Okay, so I'm looking at an image here, and I've examined it pretty thoroughly. Okay, so as you know, the human chromosome 1 is the largest chromosome in the genome. 2 is pretty close, though they're massive chromosomes. But these internally clustered telomeric sequences, if you look to especially chromosome 21, look to chromosome 16, and there's a color key for these scores if you're looking at least a similar image that I am. Green is 50 to 80.
Yeah.
Pink
Well,
is
okay.
80 to...
It's a little bit weird that like we're doing this on an audio only podcast, so I don't think like describing pictures is really the best format. But this topic
I'm just responding
is
because you said that these internally clustered location telomeric repeats are found in other chromosomes. You mentioned a couple of chromosomes, but if you actually really examine it, like chromosome 21, 22, and especially 16, there's a ton of these there. And you wouldn't argue that it's the result of a fusion event, would you? Any of these chromosomes?
well, fusions are not all that rare. I mean, like fusion events like happen decently often. Like lots of like closely related species have different numbers of chromosomes due to a lot of like fusion in its history. So it's not a rare process by any means. And it doesn't have to just be end to end. I mean, you can have like a telomeric fusion that's not end to end, and it's not going to create that head to head sequence like we see specifically in chromosome 2, which is the only place where we see that specific head to head sequence.
I'm not sure if you have access to your messages, but to be fair, I did send you the image that I'm talking about. If you don't have the opportunity,
Well, our audience cannot see it. That's
right? No, I understand.
Like, I honestly was like, I don't know. When I saw that this was going to be audio only podcast, I was like, oh, I hope I can describe it well enough, because a lot of these things of like the fish analysis and aligning the two together, it's very visual, and I kind of wanted to show images of that, but.
Isn't that mainly just banding pattern similarities, though, with what you're talking? I mean, we're not getting a detailed analysis or examination of the proteins, the specific genes. You're going to need a little bit more than a fish analysis or to speak to. Isn't it mainly just banding patterns that I talked about in my opening statement? I went over the banding.
You're talking about karyotyping. It is different than a fish analysis. And again, I just want to emphasize that the fish analysis is the key that caused the scientific consensus to form that the chromosome 2 fusion did happen. It is the strongest line of evidence for the fusion. And like I said in my opening, I made the analogy to the two halves of a broken egg, and that perfectly align on top of each other. Like,
well, they don't, though, because 10%.
You wouldn't just say that's a coincidence.
Well, no, because firstly, this goes back to my initial argument is, yeah, we can look to the fish analysis and ignore everything that surround the 600,000 bases that surround the fusion site. We could ignore the ape specific satellite DNA that I understand it's species specific, but it's still, it doesn't exist around human chromosomes. Now that there is shared satellite DNA, I understand that, but this specific type, no. We can ignore the fact that 10% of one of the chromosomes is missing when you actually put chimpanzee chromosome 2A and 2B next to human chromosome 2. Or if you were to look at this image, and I do for the audience sake want to say that I've written about a 25-page appendix that I'll make available so people can see these images. They're pretty easy to find online, though. The chimpanzee chromosomes were looked at for these internally clustered telomeric repeats. It's funny because there's some that exist, like chromosome 20. There's some located in the middle. Chromosome 9, there's some closer to the middle. But in the human genome, they're like everywhere. And so what does that do with the similarity between human and chimpanzee genomes? I mean, what's going on there? And here's the last thing I want to say and speak to it a little bit, is the fact that these internally repeated sequences, if there's so many of them in chromosome 21 and 22, but yet those are tiny, they're only like a quarter the size of human chromosome 2, then sure, we would expect then chromosome 2 to have higher density, more of them than in some of these other chromosomes that have them, because the other chromosomes are like way smaller than the chromosome 2, right? So there would be more reason to have a greater density within the chromosome 2. But I think the fact that there's been so much study done on the functional nature of these internally clustered telomeric sequences, that tells me that they are there for a reason. Go ahead, Grayson, speak to that.
So based on the ordering of the fish analysis, we would make a prediction, right? We would make a prediction that like the order would exactly match an end-to-end telomeric fusion. So what we would expect is not just a normal interstitial telomeric sequence. We would expect specifically to see a head-to-head, like forward and reverse ordering of the repeated sequences, which is the only place in the genome where that would occur. So it's a pretty bold prediction. And then when we looked at the exact place in between like the fish analysis, right where it would be targeted for that fusion site to be, that's exactly where we see the only instance of this head-to-head forward reverse sequence. That's it. I mean, that's like, how can you possibly get around that? That's like such like overwhelming evidence. I really feel like that's foolproof. There's not
okay.
getting,
That's a fair
around
point.
it.
Yeah, that's a fair point. I think we're getting closer to some discriminatory lines of evidence. Again, that's a fair point.
And with the DDX11L2 thing, there's
Well,
another.
let's save that after, because let's finish this topic on
Okay.
Yeah, and
then
gentlemen, I'm going to jump in here really quick because you guys have been going for quite a while now, and I think you guys deserve a little bit of a break, a minute or two break, just to go to the restroom if you need to, get us something to drink, wet your whistle. I'm going to go through some announcements, and then we'll continue this. This has been a great discussion. I think everyone in the audience is enjoying it, so
okay,
go ahead and take a little break here. Yeah.
Okay, I'll be back in a minute. Thanks, Praise.
Absolutely. So I'm going to go through a couple announcements here. Tomorrow we have Dr. Grady McMurtry coming on tomorrow. He's going to dismantle evolution, refuting old Earth. I mean, every time Grady comes on, he brings some great content. And then this is going to be action-packed for the rest of the week almost. So all the way, and then the 31st, which is in after tomorrow, we have Dr. Dino himself's going to be here debating Ken Rock. And that's going to be a double header that day. In 2 days, we have a double header. So we have Ken Hovind coming on against Ken Rock, but also to Film Free is going against Snaky. Snake was right. He's coming on. And then we go all the way. We'll have like a few day break there in April, but in the 3rd, we get right back to it. We have the Marian Dogmas debate. So Pastor Chuck versus Steve Christie's coming on. Then we have Dr. Dino coming back again on the 5th of April. And he'll be going against Ian Chen. So we all know, I mean, most of the people here that have been around the channel know who Ian Chen is. So that'll be exciting as well. And we got degeneration, de-evolution. Looks like another presentation with that on the 6th. And let me get back into some of the covenant or the dispensationalist debates. We'll have David Preston coming back here the 8th of April. And it looks like, you know, we all like to see Nick Sayers and Turretin fan too. He'll be back here the 11th. So there's a lot of content coming, everyone. Hope everyone stays tuned.
I appreciate that, Praise. So getting back to this, great job moderating, by the way, Praise. With what you were saying, Grayson, with these telomeric elements that are scattered throughout the genome, I am arguing that they represent a genomic feature, specifically a functional feature. Now, chromosomes 16, 21, 22, they have specifically a lot of these repeats everywhere, basically, within their chromosomes. 2 has a lot as well. 2 has the most in terms of density. Again, 2 is a massive chromosome, though, compared to 16, 21, and 22. But you're arguing that chromosome 2, although these are scattered everywhere, chromosome 2 is the only place where we find the TTAGG and the CCATT, basically, in forwards and reverse at that location. Is that correct?
Yeah.
So, and I've done a deep analysis here. For sake of argument, we can assume it's true. But is it your argument that chromosomes 21, 22, 16, they really stand out as having a lot of these internally clustered telomeric sequences? They don't have any of the repeats in reverse?
Not anywhere near to the same like as we see in chromosome 2.
But they do exist, just not in the same number, you're saying?
I don't have knowledge about whether or not they exist, but I know that they don't exist. Like if you search for like a head-to-head sequence, like you're going to find that only in human chromosome 2.
Well, I understand that the human chromosome 2 has it in the highest density. But again, where we find these sequences, TTAGG, CCCTAA, we find them in other chromosomes, some only a few. But again, those specific chromosomes, 16, 21, 22, I think I pointed out, they have them in abundance as well. But they're also tiny chromosomes compared to the chromosome 2.
Oh, you're only talking about like telomeric repeats though, right? Are you talking about forward and reverse like right next to each other in like a head-to-head formation? Because to my knowledge, that's only seen in chromosome 2.
And that's what brings me to something curious that I'd like to
Really quick before you ask that though, I just want to
Sure.
explain that
Yeah, take
like
your time.
interstitial telomeric sequences like that we're seeing in like these other chromosomes don't have to be the result of fusions. They can be resulting from just like normal chromosomal rearrangements. Like those can create interstitial telomeric sequences.
But how come elsewhere in the human genome, these interstitial telomere sites? I've got at least 3 papers that I've examined quite extensively, and they're wondering what these are doing there, since obviously they're not representative of the end caps of chromosomes, the telomeres. And some secularists have suggested that they are involved in gene expression. But what I find curious is, although the chromosome 2 fusion site is unique in its density, fair point, Grayson, I appreciate it, but some other chromosomes have their own uniqueness, because I've found that these telomere sites, they occur in perfect tandem, which we don't really see very much in the chromosome 2. We find them occurring in perfect tandem 3 to 10 times or even more on other chromosomes that no secularist that I've found said was the result of a fusion.
Yeah,
so
just a chromosomal rearrangement.
Okay, is why that we find the tandem repeat in perfect order 10 times or more, because we only find it a few in the chromosome 2 area. So how come?
Because again, in the chromosome 2 area, you would only have a fusion if the telomeres are already very short and degraded. You're not going to get a fusion occurring otherwise. But in a chromosomal rearrangement, those can occur when the chromosome is at any level of health. So a long telomere can be like rearranged into like the interstitial sequence space.
But speak to this. Why, if consistent? So we find in perfect tandem, TTAGG. We find this in other chromosomes occurring 3 to 10, because in the human chromosome 2, you do find them, but mostly in isolation. If you just highlight the amount of times that you find the TTAGG in actual perfect order, unbroken, it's only 10 intact TTAGGG. You could just highlight them in the 798 base pair region. And the reverse complement sequences, CCCTAA, that are complete within this site, there's 43 of the reverse complement telomere sequences. Now, again, the density is the highest, but in other chromosomes where the density isn't as high as in the chromosome 2, we find uniqueness in the fact that they occur in perfect tandem 3 to 10 times or more. But you're saying this is from chromosomal rearrangement.
Yes.
Why can't that be the case for the chromosome 2?
Because the head-to-head, the forward and the reverse sequences cannot result from a normal chromosome rearrangement. There's no way that you can rearrange your chromosome to get a head-to-head forward and reverse sequence of telomeric repeats. Like, that's not going to happen with a chromosomal rearrangement. That can only happen with a head-to-head fusion.
I'm sorry. Go ahead. I interrupted.
That was my last sentence. I was just saying you can only have that with a head-to-head chromosome fusion.
But the CCCTAA, which is the reverse complement of your telomeric sequences, that is assuming that on the chromosomes 21, 22, 16, and chromosome 2, those look to be the highest density that we find. We would have to confirm that there's no CCCTAA sequences there in those regions then, right?
Yeah, you can do that. There, like I said, the head-to-head, them being right next to each other, the forward and the reverse, head-to-head, that's the key.
Right, TTAGG.
Yeah.
So within the 798 base pair region, for the audience sake, I already know you know this. If you pull up an image of it, anybody can Google it. It's pretty easy. You can see that you got the TTAGGG, but then you've also got the CCATTT within that region. Okay. And again, there's only about 10, because if you actually look at the region, you got TGAGGG, TG, right? So all of this is unbroken. And then finally, you eventually get a TTAGG. But then this is in isolation. You don't get another perfect tandem repeat until dozens and dozens and dozens of sequences later. And then again, that's in isolation. And so a lot of these, they're very broken. It's very corrupt. And the same thing goes for the CCCTAA, which comes after the TTAGGG sequences. But so you would have to argue then that these other chromosomes don't have the CCCTAA sequences present in them.
Yeah, that's what I've been saying. They don't have the head-to-head sequence that we see in chromosome 2.
Right. And I would like to see confirmation. I'm not saying that this isn't the case.
I thought you had run this yourself. You said that you searched for these sequences. Like, if you go on Blast and you search for the head-to-head sequence of telomeric
it's been done. I know I've examined the chromosomes. It's been done, but they don't specify. All they do is specify that this is the 798 base pair region, basically, and they're looking for it in other chromosomes and seeing where it gets hits. And so we find a lot of hits in 21, 22, 16, but it doesn't necessarily tell us how many are the TTAGGG versus the CCATTT. So I am curious to know if where we find these repeats in high density elsewhere in the genome, if it's only TTAGGG with no CCATTT.
Yeah, it's only in the forward or the reverse direction, not both forward and reverse in a head-to-head sequence. Like,
right,
only in chromosome 2. That's, and so like the smoking gun that proves the whole fusion happened.
If that's the case, then that is a curious point. And much like the 600,000 base pair region that surrounds the DDX11L2 area, I would say much like a detective case where you're in a courtroom and one side has this many lines of evidence, the other side has this many lines of evidence. If that really is the case, that in light of all of the chromosomes having these internally located telomeric repeats, if chromosome 2 is the only place where you find them in both forwards and backwards, then that would be a point for your side within the court case. But we'd have to examine it all because, again, and speak to this a little bit, these telomeric repeats that are found across the genome, they're associated with, according to at least one of these papers that I've read, they're associated with hallmarks of gene expression because they have the same type of transcription factor binding and gene activity that we find at the alleged fusion site that we're talking about. And so these could represent functional DNA elements within healthy genes. And in this case, the healthy gene being the DDX11L2 gene, which we can move on to discussing if you'd like.
So yeah, I want to be very clear. None of that is inconsistent with a fusion model. These and again, these whole sequence of 600,000 base pairs that you've been bringing up that are like totally not found anywhere in chimp genomes. I'm very skeptical of this claim. This is the first I'm hearing of it, so I would want to read the paper after this and see if that's actually what the paper is showing. But I will take you at your word that that is what is demonstrated in that paper. Even if that is the case, it's not inconsistent with the chromosome fusion model. It doesn't count as strong evidence one way or another. It's not differentiated lines of evidence. The same with the DDX11L2 gene. Again, it doesn't really matter if it's functional or not. Like, the consensus is that it has no known function and is a pseudogene, but it doesn't matter. Even if it is a regular gene, it is still not inconsistent with the fusion model that it would be at that location.
Okay, well then speak to this then.
Wait, I was just going to say, because of chromosomal translocation, there are mechanisms that perfectly well explain this within a fusion model, and it's not inconsistent with it.
Okay, speak to this then. And you did rebut this. I appreciate it in your rebuttal, your 8-minute rebuttal. But I'd like to dig a little more deep into it because I would support my argument that even if chromosome 2 is the only spot where you actually have the forwards and backwards repeats, the fact that we find them in such few amount, and there's at least 1 or 2 studies that say that they exist in a lot of these regions for functional reasons, therefore they're the functional features of human genes. I would support that by saying, if we were to assume these were the result of a fusion, and healthy telomeres have anywhere between 10,000 and 15,000 bases, why do we find, if we had, let's say, 2 telomeres with 15,000 bases basically fuse, that would be 30,000. Or if we had a smaller one fuse, that's 20,000. You're saying the reason why we only have 798, correct me if I'm wrong, I wrote down everything you said, but as quickly as I could. Basically, before the fusion happened, you're saying that the telomeres, the end caps of these chromosomes, they degenerated. So they lost a lot of their sequences, and then they fused when they were small. Is that more or less what you were arguing?
Yeah, a head-to-head fusion would not occur if the telomeres were at their normal length, because the telomeres protect that. Like, the point of the telomeres being there as an end cap for the chromosome is to prevent a fusion from happening. So if the chromosomes are present and functional, then a fusion will not occur.
Right.
So the only way that the fusion does actually happen is if the chromosomes are highly degraded and very short.
Okay, so if for sake of argument, then we were to imagine that 2 of your tiniest size telomeres managed to fuse end to end, that means they'd be about 10,000 bases in length. Again, for the audience sake, the site we find is 798. So obviously a lot smaller than 10,000. Now you're saying that these 2 tiny telomeres, we're just assuming that they were tiny telomeres. There's no way to really know. They could have been large telomeres.
Well, they couldn't have if they fused.
Right. Well, I'm saying now you have to degenerate them. Degenerate them from 10,000 bases to what, though? 2,000 bases, 5,000 bases, 1,000 bases?
So telomeric end-to-end fusions have been simulated in a lab where, like, they degrade the telomeres and then see what happens. And when the telomeres are degraded enough, then they stick together in an end-to-end fusion. I don't know specifically offhand at how short the telomeres have to get before they will start to be sticky and have and be able to fuse together. But I think that information would be available in the studies where we have like replicated end-to-end fusion.
Right. I'd be interested to see that because, again, I find this is a prime example of evolutionists that debate like yourself, and I appreciate that. I love to debate too. You guys want to make it sound like there's never a surprise, there's never a problem, everything's expected. Well, in my opening statement, I went over a quote from a key fusion paper where the authors literally said, "Why is the site so degenerate?" They were baffled by it. They were surprised. But if this is all expected, then why were they asking? They should have said, as expected, the telomeres degenerated, they fused, and now all we have is a 798 base pair region. Why did they emphasize their shock then, Grayson?
Who's emphasizing their shock?
Well, I'll pull up the quote again from the. So in my opening presentation, I went over a key paper on the fusion, and the author. So here's the quote. They said, if the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate? Well, how come they're even asking that question if, as you're saying, this is expected? This had to be the case.
I think what they're saying is like the actual sequence themselves, like the TTA, like the telomeric sequences themselves are degenerated. Not that like why are they so short? Why are there so few of them? They're asking about why do we see the amount of mutations that we do see there?
Well, when I continue the paper, and I'll send it to you, the title is by Fan Y 2002. Gutsy Gibbon, from what I remember, cited it in her video. Genomic structure and evolution of the ancestral chromosome fusion site. And it goes, that's a long title. After that part where they ask the question, they go into possible reasons why the site is so corrupt. Maybe it was lost over time. The regions were not subject to purifying selection, and so they were hit with a lot of mutations. So it does sound like they are trying to address the small nature of it, but also the corrupt nature of it. You're right. The fact that we only find a few of these TTAGG sections or sequences within that region, which I would argue is all there for a functional purpose. That's why there's a bit of a shock there. But anyway, speak to that a little bit.
Well, I mean, it sounds like the paper that you're quoting already spoke to it. It sounds like they postulated a number of different explanations. This is a paper you said like from 2002, so I would just wonder, has there been any follow-up in the last 20 years where maybe we've learned more about this? That would be something to research for more discussions, but my first initial guess, right, it would just be a guess because yeah, I don't know for sure, but I would be happy to do more research and see if these authors have published anything more in the last 20 years. But I would think that if a genetic sequence is not highly constrained, then it would accumulate mutations faster than a more constrained area. So, given the fact that the area surrounding the fusion site has never been demonstrated to have any documented function, we would not expect that sequence to be very highly
If I could
constrained.
interrupt you just for clarification, this paper, which I pulled up, this is, from my understanding, the key paper where they talked about the fish signals and did the fish analysis. And so if that's what you were getting your argument from earlier, if you look within the paper, now would a prediction be, based on what you're saying and what the authors of this paper say, that a lot of those sites have been mutated and changed and lost since the fusion occurred, and that's why the region is so small? Would a prediction then be that within that area, within humans, the human population, that area should be highly variable then, if it's so rapidly mutating?
Yeah, I mean, if it would be if there were a lot of mutations there, I mean, some of them could have been fixed in the last 6 million years. I don't know, but I mean, if it's a non-constrained sequence, then we would expect it to be subject to genetic drift.
I have done a look. I've looked into that, and again, there, as you know, there's a lot within the technical literature. But what I found, at least, and maybe this is something we can have you look into after the fact. But that
Yeah, and I
region
gotta say
within
something real
humans.
quick, guys. You got 10 minutes. I just want to let you guys know that. Then we're gonna have closing statements.
Okay, so I'll make this quick then. From the research I've done, that area at the 798 base pair region within human chromosome 2, it's not variable within humans.
Okay,
So,
well, I would just need to see that data.
right. And
is this are you saying that you generated this data, or like
I didn't, no, I didn't generate this data. I just did. I looked into it, seeing what that region is in terms of variability. I assumed, okay, if there's any legitimacy to their reasoning why the site is so small, why it is so corrupt and degenerate that most of it's been lost or it's been subject to a lot of mutations over the last 4 to 6 million years, then I would assume the human population, there's 7 billion people, there should be some groups on this planet that have been isolated for a while, and therefore that region should be more variable. And I did not find that to be the case, but again, there's a lot of literature out there. So I would maybe push you or challenge you to look into that, and that could be a resolution on whether or not that argument fits. But since we have 10 more minutes left, I do want to respect your wish to discuss the DDX11L2 itself. And did you want to start with that? Did you want to point out your disagreements on that specific issue? You're saying it's not functional.
I'm just saying that it's very misleading of you to characterize it as this highly functional, highly important, highly critical gene when, like, I think that you're kind of talking out your, you know what, a bit to make all of those claims without any evidence for them.
Well, if you go to just look up DDX11L2, MayanLab.cloud. I don't know if I'm saying that Mayan part right. /harmonizome/gene/DDX11L2. I understand evolutionists don't trust Dr. Jeffrey Tompkins' work, although he's written some. Now, when you read Dr. Tompkins' original paper in 2013, you'd know that about 3/4 of the way in, there's a massive section on all of the genes that it's co-expressed with and how critical those genes are and genes that it's associated with and all the binding sites associated with it and your transcription start sites. And so it's curious to me that you just disregarded all that. But before you speak to that, again, look up the gene, DDX11L2 gene. It literally says DDX11L2 has 1,996 functional associations. And you've got dozens of drop boxes in all these different cell types and tissues, and you can see what it's expressed with, what it's doing. How do you explain that?
You can't see what it's doing, though. Like, you could say that there's all of these functional associations, but if I ask you just name one that shows that this transcript goes on to have any functional role of any kind, you can't show that because nobody showed that because it's never been shown.
Well, what were your thoughts on the famous 2013 paper by Jeffrey Tompkins? He did show that.
He did not show that.
I'll pull it up right now.
Yeah. So what he has shown, right, is that the area is transcribed. And he was talking about how there are two main varieties of that sequence, one of which contains the fusion site and the other does not. But again, the fusion site itself is not the promoter. The promoter is on the other side of the fusion site. So that's why, like, there are two transcripts, because there's two different promoters, one on one side of the fusion site and one on the other. I'm sorry if this is hard for our audience to visualize. There are pictures available. I think Tompkins' paper has a picture of it.
Well, it's in the first intron. It is in the middle of the gene. But I want to say this. If you go down to the paper, scroll down a little bit more than halfway.
Okay. So you know what introns, like, that they're cut out.
Intervening regions of genes. Yeah, they're cut out for the final product.
So, like, there's not much function if it's not serving as a promoter, if it's getting spliced out of the transcript. There's not really much of a chance for the fusion site, like, a section of the transcript, to have any functional impact.
But I just find that it's evolutionary philosophy and evolutionary based assumptions to disregard the fact that this gene is linked to the expression of other genes. And these genes are basically at the top of the pyramid of many important genes in cell processes. And in his paper here, again, if you
Yeah,
go
but
about
it's never been demonstrated that this DDX11L2 gene has
well, I'm trying to
any
demonstrate it right
impact.
now, but you keep interrupting as I do. So if you go about midway, anybody can look it up. Jeffrey Tompkins, 2013 paper, human chromosome 2 fusion. You could also look at the DDX11L2 gene. Just Google it, look up that source that I pointed out. And he points out clues about what type of general cell processes the DDX11L2 gene might be involved with were revealed through its detective close association with other genes. And he goes into co-expression linked to at least 3 different key genes. One, TREML1, which is a cell surface receptor for myeloid cells. He goes into TUBB1, BEND2. I mean, you can look at it yourself. And so there's, if it's a long non-coding RNA helicase gene that's producing RNA transcripts, and the site itself has 2 promoters, the second promoter being the one that actually is the genetic switch for the DDX11L2. I mean, what more do you need for function? I mean, what are you looking
You
for?
need to demonstrate function. That's what you need. So for example,
demonstrated.
So for example, with like long non-coding RNA sequences, like you brought up, you claim that this is an example of one that has function, but you can tell that we have demonstrated function for different like long non-coding RNAs. Like they have been demonstrated to have function, like you read out with gene expression or with like chromatin states or whatever. Like we can demonstrate, we can show those functions. Those functions have not been shown with the DDX11L2. So what you've been talking about with Tompkins is he's showing correlations, right? So none of this is any causative observations, or these are not experiments or anything. And I would also point out that this was 10 years ago that Tompkins first threw
Well,
all
no,
these.
I do want to.
He didn't directly show any function. He just said, hey, there are these correlations that we're observing. And he's had 10 years to try to find what the function is based on that. And he
Well,
has not
firstly,
been able to do that.
I appreciate those points. Firstly, yes, that was the original paper in 2013. He's had a paper in, I believe, 2021 where he did a thorough examination on the interstitial telomeric repeats, found a lot of amazing associations there. And he did a follow-up debunking the debunkers to people like Ken Miller, who claimed that the DDX11L2 was mostly spurious. And I want to cover that a little bit. But before I do, I find a lot of evolutionists, when looking for functions in non-protein coding genes, like your long non-coding RNA genes that produce these RNA transcripts, they don't recognize how important co-expression is, because they want to compare the expression levels in these non-coding regions to protein coding regions. But it's well known, and I've got at least 2 papers here demonstrating this, that your non-coding genes, like your long non-coding RNA genes, they're expressed at a lower level and yet functional and essential than a protein coding gene that's expressed at a higher level but has the same kind of important function. And that is why a lot of scientists, especially in health, like at the NIH, where they take this stuff seriously, I find evolutionary biologists are kind of hindering, unfortunately, the science on this. They recognize that co-expression is extremely important when looking at the functionality of certain non-coding RNA genes. And to speak to that, but I want to pull up Tompkins' response to Ken Miller.
So
Speak to that as I do.
So again, like the long coding RNA sequences that have been demonstrated to have function have easily been shown what that function is. Like we have seen it in experiments that they have an effect on gene expression. Meanwhile, the DDX11L2 pseudo gene has never been shown to have an effect on function, like on gene expression. Like you can point to correlations where, hey, early on in development in the testes, we can see that DDX11L2 is more expressed than it is normally, where its normal expression is about the level of background noise. But specifically, when you're like in early stages of development, we see a spike in the testes. Okay, so what is that? Oh, well, we noticed that normally that's the case for every like every area of the genome has higher expression in early stages of development. It's not like weird or notable or unique in any way. So again, like if you can demonstrate a specific function, which Tompkins has been trying to do for years, you think it's interesting that as his research goes on, he just kind of switches around to different topics, and he starts reading about it. He starts publishing about interstitial telomeric sequences, not about DDX11L2 function anymore. He's moved on, and now he's publishing a response debunking the debunkers. But again, he's not pushing forward his research on DDX11L2 function because he's hit a brick wall. He can't show any function. All he can show are correlations, but he has had 10 years at this, and he has had no results.
Okay, let me speak to that a little bit. So the reason why he's moved on in 2021 to the internally clustered telomeric sequences is because now that we know, which you're denying, and I know you disagree, so maybe note and respond after I'm done this, but I want to get through these couple excerpts from Debunking the Debunkers. And he pointed out, no, these critics like Ken Miller and others, mostly Ken Miller, he's the one that started this whole spurious nature of the DDX11L2. He said, no, they're wrong. The fusion site sequence binds to at least 12 different transcription factors, including RNA polymerase II, the key enzyme that transcribes genes. He said additional data in this paper, so he has it in this paper, it's very comprehensive, will also show that along with the binding of RNA polymerase is the fact that transcription has also been shown to initiate inside the fusion-like sequence in a classic promoter-like fashion. And I've been pointing out these promoters are like genetic switches. It's if you walk into a room, you can turn the light on, right? That's a switch, and you're turning on the light for the room. And so this dual promoter region, the second promoter, he's demonstrating here. I don't know if you read this paper or not. I highly recommend it.
I feel like something needs to be specified here. What I think, because I think you're conflating the fusion site and the second like promoter sequence, like the intron, because to my knowledge, the promoter is not in that fusion site. The promoter is off to one side of the fusion site, and so when the RNA polymerase binds like to the promoter and starts transcribing, because again, we already knew that this was transcribed into RNA. The issue is whether or not that RNA is functional. That's what we've been talking about. But we already knew it was transcribed. So by definition, if it's transcribed, it's going to be binding with RNA polymerase. So that was already a given. We already knew that would happen because we already knew.
Yeah, but you didn't know that there was multiple TSSs, transcription start sites.
Well, we already did talk about that. We talked about the two different modes of it. One mode does not include the fusion site, and one mode does. That's the site where like that's what we've been talking about with that secondary promoter. So we have been talking about all this stuff.
No, but you're saying that they understood that it transcribed, but basically transcribed at low levels. I'd wonder why then, when you just look up the DDX11L2 gene, it says that it's functionally associated with almost 2,000. The gene itself functions in over 250 different cell types, but it's associated with, it has almost 2,000 functional associations. Here's the last point I want to speak to, and then you can respond. Tompkins says these data also intersect, he points this out in his paper, he's got all the research there, with transcriptionally active histone marks and open active chromatin profiles that are hallmarks of promoters. I find that your evolutionary philosophy and your basic ancestry related assumptions are preventing you from acknowledging the fact that clearly there's a lot of, this is a hotbed for genome activity. No wonder why there's some uniqueness to it with your forwards and backwards repeats. No wonder why again, it's a hotbed for genome activity. It's evidence for design. I mean, I don't know how you can look at this and be like, no, this is all just spurious. It's
because it's not a hotbed of genome activity. If you look at this compared to any actual hotbed of genome activity, you'll see the numbers, and they're not at all comparable. This is very, very low on the scale of like expression. This is like on the scale of other pseudogenes, not on the scale of actual genes or actual something.
Grayson, it literally encodes several transcript variants expressed in, I've said this a few times, 250 different cell and or tissue types. That's nothing to you. That's just
Yeah. So I feel like
garbage.
this is kind of relying on people's general ignorance of like genetics and how these
He's
things
a
are
PhD geneticist. He ran a genome research center for years and years.
Yeah, but any
He's
actual
ignorant?
geneticist would understand and understand like how misleading he's being, because tons of different transcripts are expressed in tons of different cell types.
And they're functional.
No, again, that is not showing what its function is.
Well, your evolutionary philosophy is hindering you from seeing that this is function because you require genomes of junk. And so a lot of this functionality you want to chalk up to noise, spurious activity. RNA polymerase 2 is just randomly, doing its job here, latching on to, to me, I'm just hearing a lot of evolutionary philosophy.
Yeah, and gentlemen, we've gone a little bit over too. If we could just wrap it up like in 1 minute or 2 minutes, because then we have some questions we got to get to, and then also your concluding remarks.
If I may, I just want to hear Donnie's response to. I brought this up in my opening, but like the prediction about DDX11L2, right? If the fusion really did happen, then the prediction would be that the DDX11L2 gene would be near the telomeres of one of the ape chromosomes. And then when we look at chimpanzees, we find DDX11L2 right at the telomere at the end of chromosome 2A in the chimp genome. So can you talk to that a little bit? Well, like, isn't that exactly what we would expect if this was an actual fusion?
Well, I think I would have to ask a couple clarifying questions. So you're saying that the homologous gene in chimpanzee genome, the DDX11L2, is found next to its functional telomere at the end of chromosome 2A, 2B. Which one?
Yes. In the paper, I think it said 2P, which I think is 2A, but they were using P's and Q's instead of A's and B's, but I think it's 2A.
Okay, and it's associated with the same type of function that you deny basically in the chimpanzee 2A that it is in human chromosome 2?
Don't have any data about what function like it's tied with in chimps. They just looked at the sequence and saw, hey, like this is the same like sequence as DDX11L2.
Isn't the DDX family of genes, aren't they known or unique to being close to telomeres? Like, isn't this kind of
so that is another point, is that normally all of these DDX11 or DDX11L genes are, or pseudogenes. I think the DDX are genes and DDXL are pseudogenes, I think, but I could be mistaken about that. But they are normally found at the telomeres. The only time that they're found in the middle of a genome of a chromosome is in chromosome 2.
I would argue, according to my model, that firstly, assuming that the genes associated with the DDX family are pseudogenes, I am pointing out, as I have the whole debate, that no, these are necessary to sustain healthy life processes in the cell. They're not pseudogenes. And so if they are unique, though, to telomeres, existing close to telomeres, then I would argue that is necessary for their function. I mean, universally, biceps are located near triceps. I mean,
so here's something that's really interesting. And forgive me, but I didn't prepare this paper beforehand, so I'll have to get it to you afterwards. But I remember reading that there is actually like a percentage of the human population, like the DDX11L gene is not only located near the fusion site. In some fraction of the population, it's actually migrated back to the telomeres. So this kind of debunks your point about how these genes need to be in a specific location to do their function. And that's why they're in these locations, because it's all designed. Because we see like, and this is not just true for DDX11L, but there's a lots of different genes where like some people have it in this location, some people have it in a different location, and it doesn't really seem to have any noticeable impact.
Okay, I would argue that, and if you look at Contested Bones by Roop and Sanford on page 327, they do go into what you're talking about, where it says the particular gene is similar to a telomere associated gene family, DDX11L, right? Which encodes an RNA helicase protein. Like many functional pseudogenes, DDX11L2 does not produce a protein, but it does make a variety of RNAs. And it goes into basically what you're saying here with the being located next to a telomere. I'd have to see what you're saying. How you're saying that there's a subpopulation of people where the gene has migrated back closer to the telomere. If you could reiterate that.
Yeah, no, that's what I said.
Okay.
And I think we'll end it there, you guys, because we're a little bit past, a little bit, actually like 5 or 10 minutes over. So, I mean, is there anything? I mean, is there something pending you guys have to finish?
No.
Okay.
If you want, give because Grayson started. I think that's a good place to end it. Give him his closing statement. Let's say 5 minutes. I'll do the same, and then we'll do some audience questions.
Donnie, can you
Awesome.
go first with the closing statement?
Okay. Sure. Let me get to what I wanted to address here. Usually the affirmative goes first, but this has been a good debate, so I'll give you the respect and go first here. So Praise, let me know when I've got exactly a minute left. Okay.
Will do.
So here, okay. So a lot of this surrounds Ken Miller's response. Ken Miller is the one that basically said, if there was no fusion, there was no evolution. And Jeffrey Tompkins has not, I want to set the foundation a little bit for this closing statement. When it comes to the chromosome 2 fusion, whether there was a fusion or not, I'm arguing there wasn't. It is what's called an apomorphy, which means we find this trait, this unilateral mistake in humans. Okay. And therefore it would have had to occur in the human line. And so evolutionists would have to look to bottlenecks in drift to essentially explain why. I've heard Dr. Joel Duff say, well, you know, it would have occurred in a sub-tribe or subgroup. There would have been groups around the planet because random mating has not been true historically. People have basically intermarried and stayed within their tribes. So it would be very hard to fix these kinds of mistakes or changes. So basically all your other people on the globe would have to die. And then that would allow this fusion to propagate through genetic drift. And now every single person today has it. Okay. Well, if that were the case, again, that's very difficult according to the evolutionary model. But, and there's a lot of I don't knows and unknowns, even in the secular literature. I've read numerous papers in prep for this, where it's a lot of guesswork. They admit, you know, we still don't understand. We still don't understand how this propagated. They assume it happened, but we don't understand how it propagated. It's difficult. But according to the biblical creation model, fusions in general are manifestations of genome degradation and genetic entropy. And so after the flood, we actually have a bottleneck of 8 people. And then at the Tower of Babel, 500 years later, we have a bottleneck of between 10,000 and 1,000 people. And so within that time frame, if there was a fusion, it could easily propagate through the bottlenecks, but also through genetic drift. And so the creation model actually has a better way of explaining how a fusion can spread population-wide. So I'm just saying either way, it's not the greatest evidence for evolution because it's not really specific to common ancestry. Okay, they'll argue, well, it was predicted. Okay, well, what about all the dissimilarities in the region, like the ape-specific satellite DNA, the lack of homology in certain spots, the lack of homology in surrounding areas? Regardless, I just want to point that out to the audience, anybody confused. It really isn't either way good evidence for common ancestry, mostly agnostic. Now, it is discriminatory if there's not a fusion. And I think the data suggests there wasn't a fusion. Okay. And so Ken Miller has tried to make a lot of the points that Grayson has said today. And this is where I want to spend the last couple minutes going over that. So Tompkins points out, one of the main proponents of the alleged fusion site as evidence of human evolution among the general public is Ken Miller. Miller claimed that I was wrong about the fusion site being situated inside the first intron of the DDX11L2 gene based on data that excluded the 3 exon version of the gene. I then conversed with Miller via email, and he did concede that when one includes the longer transcript variants, the fusion site is in fact located inside the gene. And remember, this region, which is only 798 base pairs, it's very corrupt, degenerate. There's a lot of storytelling and evolutionary philosophy that goes into explaining, as we've seen throughout this debate, why the region is so small. The authors themselves acknowledge that it is so small, and we can make predictions. Is this region in humans highly variable? If it's not, then they can't really look to or appeal to significant loss or deleterious mutation accumulation to explain it. Okay. Tompkins continues. He attempted to explain the longer transcripts of the DDX11L2 gene by claiming that they are easily explainable by variability in the termination of transcription, so that occasionally a somewhat longer RNA is produced. This argument is completely erroneous because the direction of transcription for the DDX11L2 gene, which is encoded on the minus strand, is the opposite of what Miller is claiming. The longer transcripts begin ahead of the fusion site at the 5' end of the gene. It is a well-documented fact that transcription begins at the 5' end of genes. And you can read this whole paper debunking the debunkers. There's been no good response to the function, to these arguments about DDX11L2 gene. It functions in over 250 different cell types. And so basically, with my last 20 seconds here, the last thing I want to say is that Grayson has to answer the question, although his way is basically dodging the function. But how do you get highly functional genes by slamming together 2 chromosomes? Here's the last thing I'll say in 10 seconds. Even your cryptic centromere site, your alphoid repeats, they're also right in the middle of a functional protein coding gene, these ankyrin proteins. So what we're looking at is functional features inside healthy and functional genes, not a fusion. I yield.
Okay. Wow.
Thank you.
5 minutes nonstop. Grayson.
That was a ton of. You even brought up new points like the alphoid repeats and centromeres. We didn't even talk about that. You're bringing up new points. You brought up so many points in that conclusion.
Whenever you're ready, go ahead, Grayson.
Okay. Well, I think that Donnie is begging the question when asked to demonstrate the function of DDX11L2. He says, "Well, look, it functions in 2,000 cell types." That's literally begging the question. He has not demonstrated what that function is. He's just claimed that it has function in all these different cell types, but he is not capable of telling you what is that function because it's unknown. There is no known function. All he brings up is correlations between like when it's expressed or whatever. And again, he misspoke earlier in saying that the fusion site was an active promoter. Now that he's actually read the quote from Tompkins, it should be evident that the fusion site is simply included within the longer variant because the actual promoter is on the other side of the fusion site from the rest of the gene. So that explains why the fusion site is included, and it's spliced out of the transcript. It's completely cut out. So claiming that the fusion site somehow demonstrates function is pretty erroneous. He said that, oh, well, like, again, I've already granted, even if this pseudogene was shown to actually have function, that's not differentiated lines of evidence because like a functional gene can move to that area. Like it's on one side of the fusion site. Like we have chromosomal translocations, we have mechanisms that would explain it. It's not inconsistent with the fusion model. Nothing that he's pointed out has been inconsistent with the fusion model. Lastly, I also want to say that I found an interesting website while Donnie was talking about his conclusion there, where we can actually purchase cell strains where the DDX11L2 gene in chromosome 2 has been knocked out. So it's no longer expressed. We can actually buy cells that do not have a DDX11L2 gene being expressed, and we can see what happens. So it's only $9,350 to buy these cells to have them made by this company, Synthago. So I would just ask if anyone wants to see that. I'm sure Donnie's donations in chat are open. If he wants to spend $9,000, or maybe Tompkins can get the grant for that, because Tompkins could just easily prove that this was a functional gene by showing that when it's knocked out, here are the negative effects. It's like $9,000 on a private website, and I'm pretty sure that Tompkins is fully capable as a geneticist. He should be able to do this himself for a lot cheaper. So he could totally do this. He could show it has function. We could do a knockout test. We could totally see if these human cells are functional or if they're just totally failing because there's no DDX11L2 gene. I think that Tompkins is afraid to do this because he knows what the results will be. He knows he has never demonstrated a function for it. And finally, what Donnie was talking about with hey, maybe the chromosome fusion did happen and Adam and Eve did have ape chromosomes, and they degenerated, and then after the flood, oh, now we only have 46 chromosomes because of the degeneration, the fall of man, and Eve and the apple and all that stuff. Well, I think that's kind of funny. So he's prepared to accommodate chromosome 2 fusion into the young Earth creation model, which is kind of funny to me, that he's basically acknowledging that Adam and Eve had chromosomes that were more similar to apes than to modern humans, which is hilarious because it just sounds like acknowledging that humans evolved, or I guess devolved in his model, from more ape-like ancestors in the past, i.e. Adam and Eve. So it's very, very funny how the leaps and bounds and the kind of gymnastics that young Earth creationists have to go through in order to accommodate something like chromosome 2 fusion, which was a verified prediction of evolution and is necessary for the evolutionary paradigm. And it was validated. We have the fish analysis, which proves it. So I'll just end there. We can get to some Q&A.
All right, you guys. That was it was an amicable discussion there. I appreciate the good attitudes from both sides. I mean, it was very cordial, so I appreciate that. And it seemed to be pretty technical and comprehensive topic. So, I mean, that kind of went over some people's heads like me, but hope people check into it if they're interested into it. But now we'll jump into the Q&As. And sometimes there are these little debates within themselves.
Right. Are Q&As related to RNAs?
Yes, they are. On YouTube
Grayson and
Evolution.
we love our technical debates, don't we, Grayson? Little ERVs, little phylogenetics, little chromosome 2 fusion.
Oh yeah, I am definitely eagerly awaiting to do a discussion on genetic entropy and ERVs and all of these very niche, in the weeds kind of genetics topic.
Someone's got to debate them. That's what we're here for.
That's right.
Okay, Praise, back to you. Guess we got some audience questions. Sounds fun.
We do. We got several super chats. We'll head to those first. So whoever it's addressed to, they get the final response or the final comment. So I think we all know that. So we'll just start with Patty Smith for $10 super chat. Hi, Donnie. How do they know apes were not descendants from man, not the other way around? Lucy was discovered from a bone sticking out of the ground and supposed to be 3 MYO. This also explains C2 well.
How do we know apes today? So by apes today, I'm guessing extant apes like chimpanzees, bonobos, orangutans, gorillas. Why aren't they descended from humans? Is that what the question
How
is?
do we know apes were not descendants from man, not the other way around? Lucy was discovered with a bone sticking out of the ground and was supposed to be 3 MYO. This also explains C2 well.
I think 3 million years ago.
Yeah, I guess. Yeah.
Why apes are not descended from man? Well, I believe in separate ancestry. So you'd have your extant apes, like chimpanzees, gorillas, so on and so forth, they'd go back to ape common ancestors. Evolutionary taxonomists would call extant great apes non-human apes. And all humans today, for a number of reasons that Grayson and I have debated for hours, go back to Noah and his family at the flood 4,500 years ago. And then from there to the creation event, a couple thousand years before that, to Adam and Eve. I would look to levels of genetic diversity. You could look to orphan genes. You could look to molecular clocks. You could look to dissimilarity in some of these uniparentally inherited DNA compartments like Y chromosome. I mean, Grace and I could debate that for another 4 hours. So I don't think the data suggests that extant apes descended from humans. I think both groups are separate, not related. So
Okay, so I would answer this as saying that the same way that we know that you are the child of your parents and your parents are not your children. So we can look at the genetics and we can make that determination. I don't think anyone would get confused looking at your parents' DNA versus yours about who's the parent and who's the child. We can easily determine that based on genetic nested hierarchies, which is the same way that we know that humans evolved from like other apes, is because of the patterns of genetic nested hierarchies that we see. So it's not like apes nest within human genetic variation, like humans nest within ape genetics.
was that the question was for me, Praise? I get the last
Yep,
word.
yeah, yep.
So basically, yeah, the pedigree is a great way to determine. When you look to a pedigree, you're going to go back not to it. It would contradict what the questioner is saying, that basically humans brought forth apes according to the evolutionary model. Basically, pedigrees would bring us back to humans, fewer humans. And if you looked at chimpanzees, the same thing. Grayson, with nested hierarchies, I would argue that yes, there's a general hierarchy that exists. Humans are more similar to a chimpanzee than they are to dogs. But when you examine the details of the so-called hierarchy, which we've done, you find a lot of inconsistencies with the hierarchy. One that I'll say, for sake of time, is the Y chromosome, which I don't think the evolutionary community has given a satisfactory answer to. When you consider overall gene content, architecture, and size differences, the human Y chromosome is only 35% the same to the chimpanzee Y chromosome. But wait a minute, the chimpanzee is supposed to be our closest cousin, and it turns out that the human Y is more similar to the gorilla Y is to the chimpanzee Y. And so there's one out of many breaks in the hierarchy. So I'll yield there.
I appreciate that. And the next one is from a person we know has been around here so many times. And Stupid Whore Energy.
Oh, SWE is back.
Yeah, she's
Haven't
back.
seen her in a while. Good to see you.
And
She's
she
one of
gave
the
two of
originals.
them.
Yeah,
she's an
originals.
OG. Oh.
Thanks for the super chat support. She's going to try and stump me. This brings me back to the modern day debates, me versus SWE. What does she got?
Yeah, I think she has two for you. I mean, two separate ones. So here's the first one. If telomeric DNA is reduced or even absent, telomere proteins that bind telomeric DNA don't have DNA to bind to at chromosome ends, so they can't protect them from fusions. Why would you expect otherwise, SFT?
A good question. Telomeres, one of their jobs as being the end caps of chromosomes, are to protect chromosomes from fusing. But we live in a fallen world, and just like we have many amazingly complex and sophisticated DNA repair enzymes that are supposed to fix DNA breaks, we still get mutations. We get thousands and thousands and thousands during our lifetime in somatic cell lines, and then we also get roughly 100 that pass on to the next generation. So these DNA repair enzymes don't work perfectly, and telomeres, unfortunately, they can degenerate, and they don't work perfectly either. We see fusions in cancer cells. Those aren't healthy cells. We see fusions in the wild with equids, of course. Nobody's arguing that fusions don't happen. The question is, did a fusion happen in the human line? Now, when it comes to the humans, again, how much did these telomeres have to degenerate? Why were these authors, at least of this specific prominent paper on the fusion, why did they ask the question in surprise? Why is this area so degenerate? I'll tell you why. Because the area does not look like what was expected, or what a signature should look like if there really was a fusion. We see fusions again in cancer cells in the wild, and it doesn't look similar. It's different. It's corrupt. It's degenerate. And that's because what we find here, again, the few sequences, your telomeric like sequences, we do find that they're there for a functional reason. If this really was indicative of a fusion event, there should be thousands and thousands of these sequences, even if, and it's a guess, the evolutionists guess, well, maybe the telomeres degenerated and then they were able to fuse, and that's why it's so corrupt and degenerate. They're just guessing. They don't even know how big the telomeres would have been, because for sake of argument, if you had the smallest size telomere, 10,000 bases, that degenerated to, let's say, 5,000 bases, you're still looking at a 10,000 base signature. The signature is less than 800. It's pathetic. It's flimsy. Anyways, go ahead.
Well, you said a lot there. We basically already brought up her question in the debate itself. We already had a discussion about this. I think that she asked her question before we got there in the debate, but we already had, I mean, I told you exactly why these telomeres are so short and why that's what we would expect. And you're saying that the scientists were not expecting this, and that they were scrambling to find some kind of rescue device for why this could be. And I'll just bring up the fact that, like, the scientists in that paper are concluding definitively that the fusion did happen, that the evidence that they're seeing in their paper demonstrates that the fusion happened. They're not disagreeing with that, and they're not saying that this degradation is at all inconsistent with that model, because it's not. And, like, again, we would expect these to be degenerated because they are fused. When we see head-to-head telomere fusions, they only occur with highly degenerated telomeres. And for those people that don't know, what causes telomeres to degenerate? Aging. Whenever your telomeres run down, I mean, every single time your cells divide, your telomeres are getting shorter. So whenever your telomeres get so short that you don't have anymore, that's when you die. In fact, you can actually predict fairly accurately how long someone will live based on the length of their telomeres. If we were to measure Donnie and my telomere right now, whoever has the longer telomeres as the time of this discussion has a much higher chance of living longer based on natural ages. So, like, the older a person is, the shorter and more degenerated their telomeres are. So just having older parents is going to have, like, parents with shorter telomeres. That's just how they work.
Can I ask you a question before I have my last word?
Sure.
When it comes to, like, senescence and aging and telomere shortening as you get older, so how old was this person that experienced the fusion, would you say?
I have no idea.
So is this like a 95-year-old gets, like, and then again, there's just a lot of,
well, men can have children at any age.
So this 90-year-old man finds a woman who's fertile. How old was she?
It doesn't matter.
and then they make a baby, and then that baby has how many chromosomes?
It would be inheriting the father's chromosomal variation.
The unilateral mistake, so 46. So now we have one person, one newborn with a 46 chromosomes, based on a 95-year-old man marrying.
Well, you're assigning those numbers.
I
have never said any numbers.
Well, I mean, if you're going to look to age to basically degenerate these or shorten these telomeres,
because we know that age does do that. Yes.
So then this one person has 46 chromosomes.
Uh-huh.
How many people would you think on the globe at this time?
I don't know.
Let's just so many
because I
unknowns
noticed
in
you
the
do this
chromosome
during the debate too, is that you just tried to cast doubt on, like, how fixation works. But then in the genetic entropy argument, you use the argument that genetic drift fixes mutations every single generation. So on the one hand, you use the fixation by genetic drift to make your argument. And on the other hand, you try to make it seem ridiculous that mutations can be fixed based on drift.
Well, in the long run, if the Earth is not only, if humans don't go back only 4,500 years to the flood, then your per generation mutation rate, neutral mutation rate, will equal out to a fixation rate. But according to your evolutionary story, this unilateral mistake, the 46 chromosomes has to basically spread from this one person that now has 46. Praise it, just one sec. I'm just saying there is a lot of unknowns. There is a lot of storytelling coming from an argument that evolutionists claim is just completely solid. There's no questioning it. I mean, there's no random mating. So basically, correct me if I'm wrong, all your different tribes around the earth would eventually have to die off. And so this person has kids, and then it propagates the 46 chromosomes. It's not like people around the globe are going to die off right away, right? So eventually
everybody dies. You know that, right? So yes, everybody that was alive at that time is dead now. That's how history works.
Yeah, but everybody with 48 chromosomes would have to die. Everybody with the 46 chromosomes would have to continue on.
They all died too. I mean, everybody dies. We're talking about events in the long past. They're all dead now. Yeah.
Okay. Well, I appreciate you answering that. So I would, I almost forget what the original question was, but I would argue that it was SWE's question about the degenerate nature of it. So my last word to it, yes, we discussed it a lot during the debate. I'd like to see then where the evolutionary community predicted before. I want to see how specific this prediction is. I want them to say, you know what? And we're going to expect this fusion to be degenerate. It's going to be defunct. It's going to be corrupt. There's going to be just a few tandem sequences. There's actually going to be more chromosomes that have more of these perfect tandem sequences. It seems like there's a lot of discrepancies, unfortunately, that at least from what I've read from these papers, there's a lot of unknowns, and a lot of the authors admit to the unknowns. But then you got the YouTube debaters that are just like, no, it's all expected, no surprise, nothing to see here, folks. Just seems to be a lot of posturing to me.
I think we're going to end it there.
Right?
Well, you can give it a short, but also we'll hand it back to SFT for something very short. And we'll go ahead, Grace.
Short like those telomeres at in that 95-year-old that experienced the fusion.
Yeah, well, all of the geneticists that you're talking about that are like acknowledging like these so-called discrepancies and these gaps in knowledge, all of them say that the chromosome fusion definitely happened. That's where the evidence shows for sure. Like it's not like this is a big open question in the genetics community. This is settled science. Like this is not a currently ongoing area of like hot debate and research in the field. Like this was settled 20 years ago.
But of course, a couple things as final word. Obviously, these evolutionary scientists writing these papers on the fusion, they're obviously not going to see, well, because of these discrepancies, no fusion. And when Ken Miller said, if there's no fusion, there's no evolution, then evolution must not be true, separate ancestry. Let's see that paper get published, because they know, well, if there wasn't a fusion, then there'd have to be independent fission events, basically, with your orangutans, gorillas. Very, very non-parsimonious. It's easier to hold to a fusion. So, of course, their basic assumption going into addressing these discrepancies is what? The fusion happened. Now, why do we have these discrepancies? They list a bunch of reasons. And the question is, are those reasons valid? Are they parsimonious in light of the data? I don't think so. You think so. And that's where the debate is, basically. So I appreciate it.
All right, perfect. We'll head to the next one by SWE coming at you again, Donnie. Is Donnie aware that the members of the DDX gene are located right next to the telomere, except for the DDX11L2, which is right next to the fusion site? Isn't this evidence that this is a remnant of fusion?
Right. I think that relates to Grayson's final question in the discussion. Firstly, I'd want to look at that a little bit more deeply. The DDX family, the evolutionists are going to assume, as we've seen in this debate, are a pseudogene family. Anytime you hear the word pseudogene come out of the mouth of an evolutionist, you know that this is based on evolutionary philosophy, evolutionary based assumptions. They're assuming that, okay, these are the remnants of once protein coding genes. There's different kinds of pseudogenes, processed pseudogenes, duplicated pseudogenes. Nonetheless, it all basically means this was once a protein coding gene. It's now defunct. It no longer codes for protein. But again, we've pointed out throughout this debate that we have long non-coding RNA genes that are detrimental, that are functional and essential in healthy life processes. And this DDX11L2 gene is a long non-coding RNA gene. And the site, the fusion site, which is overlapped by this gene, and that's why it's an issue for the evolutionists. Here's the last thing I'll say, and then Grayson can go, is if it really is functional, which I think the data is pretty clear that it is, then how do you get highly functional genes by slamming together two chromosomes? I mean, if functional as we're saying, then there couldn't have been a chromosome fusion unless there was basically this DDX11L2 gene and there was two genes in the middle of it, maybe. And then they got deleted somehow and then brought the gene together to just magically overlap the so-called fusion site, which has multiple transcription start. It's a hotbed of genetic activity. And I understand Grayson wants to deny it, but maybe this DDX family, this DDX11 family, maybe they, in order for their function and their functional associations, need to be near functional telomeres. Just like humans worldwide have their biceps near their triceps, have our skull with our brain. But the claim that the DDX11L2 gene is not near a telomere, but every other one is, I'd have to look into that and see how valid that is. Go ahead, Grayson. You can clarify.
Okay, yeah, it definitely is. There is no other DDX11L gene that is located like not next to a telomere. They are all exclusively located near telomeres, and so is DDX11L2 in chimps. The only time that DDX11L gene, like pseudogenes, are found not associated with telomeres is at the fusion site in chromosome 2. Donnie brought up a whole lot in that topic, but I can assure you, like, you can go look this up. Look at the main paper by Costa et al. From 2009 that goes into all you could want to know about DDX11L genes. They are all located at the telomeres except in this one single instance, which is pretty strong evidence. Also, he says that this is like the third time that he's asked the same question. I've answered it every single time, and he keeps asking it, which makes me feel like he's either not hearing my answer as an answer to his question or what. I don't know. He's finding it somehow not satisfactory. But I've explained this. This is like the third or fourth time now that I've answered this question about how a functional gene can result from a chromosome fusion. And again, I'll just grant him that it's a functional gene. I don't think I have to. I think it's very clearly demonstrated that this is a pseudogene and not a functional gene. But even if I were to grant that this is a functional gene that's located in this region, that is not inconsistent with the fusion model. Like I have brought up numerous times, chromosomal translocations happen, okay? And the actual like meat of the DDX11L2 pseudogene, the majority of the gene itself is all located on one side of the fusion site, right? Exactly where it's located in the chimp chromosomes. Like I showed that as well. That's in that Costa et al. Paper from 2009 too. That DDX11L gene is right next to the telomeres in chimps. It's right on one side of the fusion site in humans. And the only thing on the other side of the fusion site is a promoter. That's it. It's not like there's anything else that's related to the DDX11L pseudogene on the other side of the fusion site except for a promoter. Just one promoter. That's all that Donnie is saying that we can't explain. How can you possibly get a promoter on one side of the fusion site if there's a fusion? Like chromosomal translocation. It's super easy. This is not a discrepancy. This is not an inconsistency. This is not anything that is even difficult to explain in a fusion model. There's nothing contradictory in this question.
Okay, so I appreciate the response. As a last word, like I noted at the end of our conversation, due to limited time, Roop and Sanford, Dr. Sanford, they address this page 327 on the chromosome 2 fusion, chapter 13. So they do admit, at first, this might seem to support the fusion hypothesis, since the reputed fusion site is found close to a gene typically seen near telomeres. However, we need to look more closely. A similar family of genes is also found in the chimp genome. While these genes are also generally found toward the ends of chimp chromosomes, they are separated from the telomere by a sizable amount of intervening DNA. So if we assume that the DDX11L2 gene on chromosome 2 came from an ape fusion, it should be separated from the telomere-like fusion site by this large ape-specific tandem repeat. Absence of this intervening DNA between the DDX11L2 gene and the reputed fusion site is not consistent with the idea that a fusion occurred in an ape population. Now, they don't rule out the possibility, like I said, of a fusion event directly in the human line. And maybe that's why there is that lack of genes surrounding the fusion site. Maybe that is why there's a lack of ape-specific satellite DNA, because maybe there was a fusion in the human line, unrelated to, and even your alphoid repeats, we'll have to do another debate on this and talk about that. Those are totally different type. There's no homology when you look at the alphoid repeats or sequences in chimpanzee chromosomes. So yeah, Grayson can focus on the points that seem to support his position and ignore all the dissimilarities. But I'm saying, like a detective case, like in a courtroom, when you consider everything, it is the creationist side that has the upper hand. And so I'll yield there.
Awesome. That was a very comprehensive response. I appreciate that, guys. So now we have Doki with, he's sharing a piece or a slice of pie with you guys. So thank you so much, Doki. And well, now we have a rival to SWE now. Her name is Tanya Brown. So she has 1, 2, 3, 4 questions. For I think 2 of them, okay, one of them is for Grayson, so at least we have that. So praise question. I'm not sure if I understand. Is saying the fish comparison is 100% the same, just like saying chimps and humans are 99% similar, only true if you ignore some parts of the genome?
No, I think Grayson's on mute.
Too?
I think is that for Grayson?
No. Yeah, there is no specific person.
I think because Tanya Brown, I think, is a creationist, so I assume this was a question for Grayson. But if it's for me, that's fine. You'll have to rephrase it though. Go ahead, or re-say it.
I'm not sure I understand. But like they say, is saying the fish comparison 100% the same, just like saying chimps and humans are 99% similar? Only true if you ignore some parts of the genome, question mark.
Oh, okay. Well, it sounds like it's more directed at Grayson. So let Grayson respond, then I'll go. To be fair, Grayson deserves some final words here and there too. So Grayson, consider it your question, and that way you'll get the final word.
I'm sorry. I have no idea what this question is asking.
I think I understand what it's saying. Is the fish analysis assuming based on ignoring large portions of the genome? I will just respond with, I don't know, but I would respond with, regardless, banding patterns, the fish analysis, synteny, homology. This is all just very minor aspects of this debate, because we have to weigh the dissimilarities with the similarities. And yes, there are some points, admittedly, to be fair to Grayson, there are some points that seem to suggest there is a fusion, but I am arguing there are many more points, discriminatory points, that say there wasn't a fusion. And just like any good court case, that would rule out or overturn there being a fusion. And you can look to not just the Fission analysis or the banding or so on and so forth. Again, I pointed out the alphoid repeats, the fact that there's no good homology within the alphoid regions. There's a publication by Half and Willard, and they stated because of this lack of homology, basically, in similarity with the alphoid repeats, they said this implies that the human chimpanzee sequence divergence has not arisen from a common ancestral, and then it's a specific satellite repeat, because the human alphoid repeat DNA sequence does not closely match homologous chimpanzee centromeres and chromosomes. You got the lack of specific ape satellite DNA. The site itself is corrupt. It's degenerate. The regions outside the fusion event, they're not found in chimpanzee. These are important genes. And so when we consider everything, it's a cumulative case, as the evolutionists say. They always say it's a cumulative case. And so I am arguing tonight it's a cumulative case, that the most parsimonious explanation is there was no fusion. Too many discrepancies. Go ahead.
Okay, right. So just the fish analysis alone proves that the fusion happened. Not a single line of evidence that Donnie has brought up the whole night has been inconsistent or contradictory with a fusion model. So the preponderance, the weight of the evidence is firmly on one side of this debate. There's a reason why Tompkins is the only geneticist in the world, basically, who disagrees with the chromosome 2 fusion.
You can give
Make it pithy, SFT. We're going to get to the next one here.
No, you give him the last word. He deserves it.
Okay. Or I mean, yeah.
Yeah, it was a question for me, even if I didn't understand it. I'll just say, like, if the idea is that the fish analysis is leaving like huge gaps, yeah, I think that there's only a certain number of like fluorescent proteins. I don't know if they're using like 30 of them or whatever, but they're just going to be binding like in a certain order. So it's not really that you need full 100% coverage. It's more of like overall that the order should be the same as to how these proteins are binding. So that's the key to the fish analysis.
Right. I'm not sure if it's specific to the fish analysis. I just know certain portions, mostly synteny, and then also the cryptic centromere comparisons. The secular papers even admit that you can, when you're looking at the data and these databases and doing these analyses, there's a lot of meaningless ends, basically showing that we don't have good data. And they also use the human genome as a scaffold. And so a lot of these questions, especially with synteny, I'm not 100% certain on the fish analysis, they are yet to be resolved until we have better sequencing data on chimpanzees. But I like to say, for sake of argument, who cares? If there is some good synteny, shared gene order along chromosomes and chromosomal organization, again, I've got at least 5 papers showing that synteny is the way it is for functional purposes.
Well, this ultimately just gets into the discussion of junk DNA, where like you have the religious position that all DNA is inherently functional, and like I'm just going with where the evidence shows. If a portion of the genome can be demonstrated to have function, I don't have a problem with calling that functional. If there is no function that can be demonstrated, then it cannot be said to be functional unless we can demonstrate that. It's, innocent until proven guilty. So you got to prove that it's functional before you can say it's functional. I think that's pretty fair. So we can move on now.
Yeah,
well,
we have another. Okay, go ahead, SFT.
No, you know what? Give him the last word. Move on.
Okay. So this is for you, Grayson, from Tanya again. If the head-to-head nature of the supposed fusion is what makes it so convincing as fusion, on what basis do you say fusion is common when you claim other fusions aren't H2H?
On what basis do I claim that fusions are common? Because they are common. Just like head-to-head fusions may not be common, but fusions in general are pretty common. You can look at like closely related species, like species that like look very similar. And you can compare the number of chromosomes that they have. And like, even though overall their genetic similarity is very high, the number of chromosomes that they have can be very different. Like you can see evidence of like multiple fusion events, like in species like past genetic history, even in a creation model. Like if you believe like that everything that we have today, if like in microevolution or whatever it is that the young Earth creation model has, you'd still have to believe that there were a ton of chromosomal fusions that occurred since the flood in the time of Noah to result in what we have now. Like there are like canine species that have like way different numbers of chromosomes, or like horses, or like stuff that most creationists think came from like one common ancestor on Noah's Ark, that show evidence of like having numerous chromosome fusions. So like I think that both models have to acknowledge that chromosome fusions occur and are not rare.
Both models do got some explaining to do. And so I'll say, yes, chromosome fusions do happen in nature, but typically those fused chromosomes are deleterious. They reduce fertility. The chromosomal architecture is disrupted. And therefore, most of the time, selection is going to work against those kinds of events. And from my understanding, now I haven't looked at every single paper, so I don't want to say all, but most, which means this would be an exception. I wonder if Grayson can speak to this. Most fusions, at least in mammals, that we've seen in nature involve satellite DNA at breaks at or near centromeres. And only telomere to satellite DNA or satellite DNA to satellite DNA are found in chromosomal fusion sites of living animals found in nature, not telomere to telomere fusions like we see here in the human chromosome 2, as purported, would be a telomere to telomere fusion. And so if it really is a fusion, it's an exception to the rule. Now, some have said all known fusions in extant mammals involve satellite DNA, but again, the technical literature is so wide. I don't know if we can determine that. So even if we said most, it still makes this so-called fusion an exception. Can you speak to that a little bit, Grayson?
Yeah, so what you're saying is not wrong. Like the vast majority of fusions that we observe, like the most common type of fusion, is not a telomere to telomere fusion that we've been talking about all night with chromosome 2. So the telomere to telomere fusion is a lot more rare. I don't think that it is unprecedented. I think that like if I'm remembering correctly, there are studies with like horses or with pigs that have shown, like just like with chromosome 2, like evidence for a head-to-head telomere fusion in that species past. So I don't think it's unprecedented, but it definitely is not the most common form of fusion, and it is more rare. So that's for sure.
I would say that telomere to telomere fusions, there have been some cases that I've found where they are in human cancer cells. But I mean, we'd all agree those are not healthy cells and are obviously connected to genomic instability and disease, of course. So that's not going to do much for a telomere to telomere fusion like we might see in the chromosome 2 fusion. I've heard about the pig and horse examples possibly being telomere to telomere fusions. Those might be inferred. I know some have backed away from that. And so the jury might still be out on that one. But either way, I think we could agree it would be an exception to the rule. That doesn't necessarily completely refute it. But I think when we get into the details, that's where the major discrepancies lie.
Yeah, so just for the final word on that, I'll say like I wouldn't necessarily throw away the cancer examples, because like they're showing us mechanistically how this is possible, and like it doesn't end the cancer cells or whatever. They continue to reproduce after like telomere to telomere head-to-head fusions. And like the example of why it would be in cancer cells is because those would be examples of like where you might have some it would be more likely that a telomere to telomere fusion would occur if there's like, highly replicating cancer cells. So I think that the cancer cell example can show mechanistically how these things can occur when you have conditions of highly degraded telomeres. So that's the last I'll say on that. We can go on to the next question.
So another question for you, Grayson. If we didn't know the sequences of humans, apes, or our supposed common ancestors, why predict fusion and not duplication?
Because it's more parsimonious that there was one fusion event rather than like multiple duplications in all the other great apes. And again, we don't need to know the genetic sequence in order to do something like a fish analysis. You don't need like specific knowledge about what the genetic sequences are in that case. You're just, again, just looking at the order of these glowing, like staining proteins. So, yeah, you don't need to know the genetic sequence in order to prove the fusion. And the fusion is more parsimonious because only one fusion needs to occur instead of needing multiple duplication events. So it's just a, Occam's razor.
Praise, can you reiterate the question? Can you say the question again?
If we didn't know the sequence of humans, apes, or our supposed common ancestors, why predict fusion and not duplication?
So when it comes to the human chromosome 2, they didn't expect the fusion until we had the chromosome numbers. 46 for humans, 48 for the great apes. And then they figured, well, it either has to be independent fission events to give your but basically everyone would have had 46 chromosomes, right, Grayson? And then eventually through these independent fission events.
Yeah, like for it to be duplication, like the common ancestor for all the great apes would have to have 46, and then humans just maintain that number, and then all the other great apes had a duplication event that made 48.
Right. So a lot of rightfully, even in the evolution community, have rightfully pointed out that that's just it's kind of a convoluted way of explaining it. Or a lot of times the most simple answer is the right answer, and so to them the fusion event made more sense. But for many of the reasons why in the last 2.5 hours, I think there's pretty good evidence to be made against a possible fusion. But for sake of time, no point in going through all that again. So appreciate the question.
And you can finish it out there, Grace, if you have someone else to say that.
No, we can just move on to the next question.
All right, this is the last question of tonight. This is for you, Grayson. Again, if they prove function, would that convince you it's not a fusion site?
No, as I already said, like there's nothing inconsistent about having a functional gene at that location. Like there's nothing that would contradict the fusion model. It's not differentiated lines of evidence. Like if it was demonstrated that the DDX11L2 pseudogene in fact did have a function, then I would acknowledge that it was a functional unit of the genome. Like I see nothing like that. I don't have any like reason why I would prefer it to be non-functional or have function. I don't have a preference either way. If the evidence demonstrates that it has a function, then I would say that it's functional. As of right now, we do not have evidence that shows us what its function could possibly be. So I'm not going to say that it's functional. But again, whether it is functional or non-functional is not related to the fusion. The fusion model...
If you don't mind, I'm sorry to interrupt. Can you just reiterate your explanation? You don't have to take a whole long time. I know we're both getting tired, because I know you said you answered my question in detail about how you can get functional genes by slamming together 2 chromosomes. Can you reiterate that explanation again real quick?
The short of it is chromosomal translocation.
So chromosomal translocation is going to take a pre-existing functional gene like DDX11L2 and move it over the fusion site.
It can happen. Yeah, this is like the mechanistically possible. There's nothing that would preclude that from happening. Like we know chromosomal translocations do happen. Genes can move around. And also,
where has that been? It just seems like
real quick. I sent you in the private chat. I know that the audience can't see this, but it's an image of all the various DDX11L genes and their locations on the different chromosomes for chimpanzees, different kinds of apes and for humans. And so you can see that most of all of them are located on the ends, and you can see that there are two human variants, one of which has the DX11L2 gene located at the fusion site, and one has it near the telomere. So there are two populations of humans.
But what about the dissimilarities? What about the intervening ape-specific satellite DNA that's not there around humans, but it's there in chimps? You just keep focusing on the
it's there in chimps, but it's not there in bonobos, which are the closest related ancestors. So it's
not there as much in bonobos, or it's completely gone in bonobos?
It's different satellite DNA. The sequence is different. It's species specific. So chimps have their own that they evolved. Bonobos have their own satellite DNA that they evolved. Like this is something that happened in the chimp line after it split off from the human common ancestor.
I would be very much interested to see the evidence for the bonobo ape-specific that type of satellite. To be clear, all of your great apes, actually Lorraine Drosophila in the chat, good to see you. She said that I claimed that humans are related to the great apes in my opening. No, I think you caught this, Grayson. I was just steel manning the evolutionist position. I was saying, according to evolutionary taxonomists, humans are apes, and the other great apes are non-human apes. And so when the evolutionist says all great apes share a common ancestor, they're including humans. For me, I'm not including humans. So that wasn't me. I just wanted to go on record. That wasn't me saying I now believe humans and the great apes are related. But when it comes to the ape-specific satellite DNA, all your great apes, including humans, have lots of similarities in different types of satellite DNA, but there is one specific one that humans lack, chimpanzees, gorillas have. But you're saying bonobos have a completely different type there. I'd be interested to see that.
Yeah, but in great apes, it is the satellite DNA is species specific. Every species has their own unique satellite DNA.
Well, wait, the gorillas and chimpanzees have the same.
No, they're unique.
No, come on. The Ventura paper says humans lack that which gorillas and chimpanzees have. That line wouldn't make sense.
Well, I don't know what to tell you about that line, like quote mine from that paper, but I'll like you might say that the amount of satellite DNA might be similar between gorillas and chimpanzees, but
that's what I'm saying. That's what I'm saying.
But the sequences themselves are species specific.
When I read through the Ventura paper, Gutsa Gibbon in her video, she made the argument it's species specific. My understanding of it, but maybe we got to dig deep into it, is humans lack this specific satellite DNA, but your chimpanzees, gorilla, very large comprehensive paper. Okay, maybe I'm misunderstanding it, but not just the Ventura paper. There's another paper too that goes into the species specificity of this type of DNA. And it sounded like they were saying bonobos lack the same amount as chimpanzees do, but not that it's totally different. Because if chimpanzees and bonobos go back to a common ancestor, then that common ancestor would have provided them that specific satellite DNA. And it sounded like it was saying the bonobos changed enough where it's a little different than the chimpanzee one, but it's still there. But humans not there at all. Does that make sense?
Well, humans do still have satellite DNA. I mean,
right, but not that specific type.
Yeah, again, the amount of satellite DNA varies, and it can be similar between chimps and gorillas, but the actual sequence of the satellite DNA is going to be species specific. But the sequence itself is going to be more similar in chimpanzees with their closest related ancestors, bonobos, than it's going to be with gorillas, which are more distantly related. So I forget what even the original question is, but like my point is basically, this is what I've set out to do tonight. Hopefully, I've done a manageable job, but I've tried to establish that all of the puzzle pieces fit together in a way that like would test the disbelief that it could be a coincidence. Like the puzzle pieces all fit together too perfectly for it not to be a fusion.
So I would say the same thing, basically. We'll leave it up to the audience. I would say that the puzzle pieces fit together in a way that suggests there wasn't a fusion, but there are some points of data, okay, I want to be objective, that seem to support a fusion, but there's more points, too many discriminatory lines of data that suggest there's not a fusion. The synteny, I don't believe works. The homology doesn't work. What is interesting, but I've got these pictures laid out, just like you pointed out, Grayson, where we've got the human genome, their chromosomes lined up with the chimpanzee genome and their chromosomes, but specifically the alphoid repeats and also the internally clustered telomeric repeats. And there's massive differences. They're almost non-existent in the chimpanzee genome, and they're scattered out around the human genome extensively. That's interesting. Those are massive differences. So my point, and I think the audience gets it, is where you're focusing on the similarities that seem to help your case, you're ignoring a lot of the dissimilarities and invoking a lot of storytelling to explain those dissimilarities. When I'm saying, no, those dissimilarities mean that the most parsimonious answer is the fact that there wasn't a fusion. I'm trying to focus on those discrepancies that I'm not finding very convincing explanations from the evolutionists from. And I'd like to see from you your explanation of chromosomal translocation to explain the DDX11L2 gene basically just moving over nicely this fusion site. I mean, you're saying that it can happen, but I want to see like a paper where this happened. That's pretty unbelievable.
Specifically for the DDX11L2, I brought up the fact that it exists, that gene is in the chimpanzee genome, and it is right where we would expect it for the chromosome. It is exactly where we would predict it to be. It is right at chromosome 2A, right next to the telomere that fused together, which it perfectly explains why DDX11L2 is at that location next to the fusion site in chromosome 2. So it is within our prediction exactly. The only thing that is on the other side of the fusion site is a promoter. That's it. So the only thing that I would need to explain is how you can get a promoter to move, which happens all the time. You acknowledge transposons exist. That answers your question.
So you're saying the promoter move,
translocation happens, then that answers your question.
What move? The promoter, the DDX11L2 gene, or both?
The promoter, just the promoter. The DDX11L2 gene does not need to move at all because it's in the exact same location in the chimp genome.
But again, if it's highly functional, which I am arguing it is, and I think the case is pretty strong, since it's literally an alternatively transcribed, post-transcriptionally spliced, post-transcriptionally regulated, and network-connected expressed gene, okay, how is that supposed to arrive by a head-to-head fusion of? It definitely wasn't there before.
Gene, is it?
It is.
But definitionally, it is not, because a gene codes for a protein. If it's not coding for a protein, it's not a gene. It's not a gene. It's not a functional gene. So just definitionally, it's not.
Network connected express gene. So your DDX11L2 is associated or helps regulate the DDX11.
But it doesn't, though. We know that.
What I want to point out before I forget. Okay, so here it is. The DDX11L2 gene produces RNA that helps regulate the DDX11L gene family, as has been shown with other functional pseudogenes. Dr. Tompkins has shown that. And you can also look. I
don't think that he's shown that.
Wait, you can also look at the gene itself. Again, almost 2,000 functional associations, just all meaningless to you. I mean, come on, your evolutionary philosophy is leaking out of your nose, out of your ears, out of your sweat glands. I mean, this is a functional gene.
Tompkins is the only scientist that holds that opinion.
Wait. No, Praise. I want to say the gene, just look it up. DDX11L2 gene. He didn't write that database. It says from a number of databases, this gene has 2,000 functional associations. That's not from Dr. Tompkins. I looked that up myself. That's right from the secular literature. Yep. So that's nothing to you?
It's not nothing.
If it was
The
5,000
thing
functional
is
code,
that that's not unique. Like there are tons of different transcripts that will exhibit similar amounts of associations.
and they're
And that
functional.
doesn't mean that it's actually doing functions at any of those sites. It's not carrying out any known function that we can know. Like all I have to do to debunk you on this is just to ask you, what is the function? You're telling me, oh, it has a function here, it has a function here, it has a function there. But if I ask you what that function actually is, it's nothing but crickets.
No. Okay, Dr. Tompkins literally says this. What might this DDX11L2 gene be doing? And he answers it. He answers your question. He says, my research showed it's expressed in at least 255 different cell types. It's also co-expressed, turned on at the same time with a variety of other genes and connected to processes associated with cell signaling in the extracellular matrix and blood cell production. And then he says the fusion site is itself functional and serves an important engineered purpose. We've talked about how it's a switch, a promoter that you say move there.
It's not though. That's not the fusion site though.
The site, the fusion site itself, is the second promoter of the DDX.
No. That's mistaken. The promoter is on one side of the fusion site. It's on the opposite side of the fusion site as the gene itself. So the promoter is not the fusion site. The fusion site itself is not.
Are you saying the fusion site is not right in the middle of the gene?
The fusion site, on one side of the fusion site is a promoter, and on the other side of the fusion site is the gene itself.
No. The fusion site is in the middle of the gene, Grayson.
Yeah, because one promoter is on one side of the fusion site. That's the only reason why you would say
Tompkins demonstrated with the short transcripts, with the long transcripts. You can look at the diagram that he laid out. I mean,
the short transcript does not involve the fusion site at all, right? You acknowledge that, correct? The short transcript. The long transcript does have the fusion site transcribed. The difference is that when we look at DDX11L in chimps, there's no long transcript. There's just the one version. And it's just on that one. It's just in the genetic sequence on the one side of the fusion site. The only difference in humans is the one promoter. That's it. Like the fusion
site is in the middle. Well, I've explained this. But Grayson, the site is in the middle of the first intron.
Yeah, introns are cleaved out. The only reason why it's in the middle of that first intron is because that promoter is there. If the promoter wasn't there, there would be no second version. There would be no long version. The only reason there is a long version is because one promoter happens to be in that location on the other side of the fusion site.
But that's the whole point, is these long non-coding RNA genes, they function almost exactly like protein coding genes. They got the exons, they got the intervening regions like the introns that are cut out for basically the final RNA product. In this case, I mean, this is all
prove that those have functions though. This we cannot prove.
It all has. It's your evolutionary science that denies the genome-wide activity. Let me ask you this question as we wind down, because we've managed to go 3 hours. I feel like you've avoided answering a lot of these major dissimilarities, such as just compare your human genome, all the chromosomes, the chimpanzee genome, all the chromosomes. Even when you line up 2A and 2B, put them on top of each other next to the chromosome 2 in humans, 10% of it's missing. But literally, genome-wide, there's massive, massive dissimilarities in your alphoid repeats and also your internally existing telomeric sites. How such dissimilarity? Like, was there that much rapid evolution taking place after the split?
I don't know if I would say rapid evolution, but yes, they're like, these are clearly two different species. I mean, humans and chimpanzees are two different species. They're going to have genetic differences. Like when we compare the total like base pair sequences, humans are still the closest to chimps out of any other species.
Well, the human chromosomes, I just wanted to point this out. Human chromosomes 2, 16, 21, and 22, they comprise a massive amount of the fusion site sequence over the length of their entire landscape, euchromatic landscape. So I'd be curious.
Well, it's not the fusion site sequence now, is it? Because it's not head to head.
Right. And that's your claim. And I'd really love to get a verification of that, because that's,
You can verify it yourself. You said that you ran the different blast sequences.
no, I didn't
like
ran
in
the, no, I didn't run the blast clears. You can take the images of the human genome, the chimpanzee genome, and you can see each chromosome. You can see how many of these internally clustered telomeric sequences exist on each chromosome, and it's all color coded. A lot of people have already done the blast analysis, and so these are the visuals from it. And to me, it's pretty clear that these fusion site sequences are in abundance on several of these chromosomes, but I don't
That's
see
not
anywhere.
head to head, though, is it? You're just looking at telomeric repeats. That's all you're looking at. You're not looking at, are they head to head? Is this a fusion site, or is this just interstitial telomeric repeats? There's a difference.
Well, given how degenerate, defunct, and corrupt the fusion site is, I don't think it'd be much of a surprise that you're going to have some of those reverse complement sequences. Since DNA is double-stranded anyways, I don't see why we wouldn't find those on, let's say, chromosome 21, 22,
Well,
16.
then show it to me on any other chromosome other than chromosome 2, and I will publicly say that Donnie was right. But until you can show me that, then you're not right.
Okay. I would just challenge you the same thing. Show me that these chromosomes that are literally, I mean, just look at the picture. I sent it to you. You look at it after. Human chromosomes 2, 16, 21, 22. I mean, they're just peppered with this fusion site sequence. Now you're
But
saying
it's not the fusion site sequence. As I keep
because you're saying
harping
right, you're saying the CCCATT is not there, but we'll look at that after. Praise, go ahead. Grace and I appreciate the back and forth.
Yeah, well, that was the final question. So if you want to shut it down, SFT, or how you want to do that, that's just let me know. And then yeah, it was an interesting debate discussion. You guys are well endowed with that technical stuff, so I appreciate that.
Well, Praise, I appreciate you moderating this podcast debate, episode 2. I think both episodes so far have been, personally, a couple of my favorite debates, actually. Last one with Snake was genetic entropy, tons of fun. This one here with my man Grayson, coming well prepared. This was, it's not a lot of fun when you discuss these technical topics with people who don't know much about them. But Grayson comes in prep. This was an excellent discussion. I really enjoyed it. 3 hours just on the chromosome 2 fusion. So there's not all that much out there, I noticed, in the world of YouTube on the chromosome 2 fusion, minus a few videos that both of our communities have done. And so now we have an extensive debate that people can look to, enjoy, watch, consider both of our points, and come to their own conclusion. That's why we do these. Critical thinking is important. Let's jump into the octagon. Let's debate these issues. Why only exist in echo chambers? No, this is what I like. This is what it's all about. So Grayson, I'm really glad to have evolutionists like you around in this community that are always up for a debate. Grayson's never turned down a debate, which means we'll probably have another 20 of these. So that's my last word. Grayson, last words from you.
Did you say I'm well endowed with technical information? Is that how you described me and Donnie?
Well, I mean, you guys
I would just like to clarify, it's 100% true. Ladies, I'm very well endowed with technical information.
And
anyways, I just want to say,
what was that word you used earlier? Amicable? Very
amicable, baby. It was amicable.
Praise has his thesaurus with him tonight.
I do.
So yeah, I do. I love these debates. I love these nerdy technical debates. I love, especially on genetics. It's a great topic. I want to call out C Science. He's in the comments, and he was saying, you got to get me on to discuss Lucy. I would love to have a debate with C Science on Australopithecus and Lucy. I think that'd be very fun. It's a topic that I haven't done a debate on yet. So yeah, I would love to debate Australopithecus. I'd love to debate genetic entropy, junk DNA, ERVs. Bring them all on.